Good afternoon. My name is John Draper, and I am the Vice President of Research and Training at the Stem Cell Network. And it is my pleasure to welcome you to the Canadian Rare Diseases Ecosystem Symposium, Genetic Therapies for Rare Diseases, on behalf of Stem Cell Network and the National Research Council of Canada. While we meet today on a virtual platform, I would like to begin by acknowledging the Indigenous peoples of all of the lands that we are on today. The work of Canada's Stem Cell Network and the National Research Council of Canada occurs from coast to coast and on the traditional ancestral and unceded territory of many Indigenous peoples and nations, including the Anishinaabe Algonquin Nation, where SCN and NRC are headquartered. We honor and recognize and respect these nations as the traditional stewards of the lands and waters where we work and meet. Many of you will have some familiarity with Stem Cell Network, but for those who don't, Stem Cell Network is a Canadian not-for-profit that supports three main objectives. We fund stem cell and regenerative medicine research, we train the next generation of highly qualified personnel, and we catalyze the knowledge mobilization and transfer of stem cell and regenerative medicine research. For more than 20 years ago, and supported by the Government of Canada, SCN has invested over 148 million in Canadian regenerative medicine research, training, and outreach to date. And so on to today's event. We're extremely excited to be hosting this in conjunction with NRC. This is a national event which will afford a chance to bring together an excellent lineup of speakers and panelists who together will explore the Canadian perspective for the clinical translation of rare disease gene therapies. The importance of rare disease targeting gene therapies that are now either under development or are transitioning to the clinic cannot be understated. These treatments represent lifelines for patients who endure the daily chronic and are all too often life-threatening burden of a rare disease. SCN recognizes the important contributions our research community can make in this area. So since 2019, we've invested over 5.8 million in rare disease research across 19 research projects at various stages along the translational pipeline. Today's event promises to offer insights into recent advances in genetic therapeutics, the opportunities they offer, and the challenges that need to be addressed in Canada to bring new rare disease treatments from the bench to the bedside. And so with that, I will now welcome and hand over to my co-host, Dr. Freya Vakur-Turan, Program Manager and of the Cell and Gene Therapy Challenge Program at the National Research Council of Canada and Adjunct Professor at Concordia University. Freya, over to you.
Thank you, Dr. Draper. Bonjour à toutes et à tous, and welcome everyone at this first session of a series of symposia that are jointly organized by the Stem Cell Network and the Cell and Gene Therapy Challenge Program of the NRC. First of all, a big thank you also to the other members of the organizing committee, Drs. Philippe Campo, Will Costain, Jim Dowling, Lucie Germain, Joseph Mancini, Kelly Parato, Etienne Richer, Cynthia Shippen, Rizini Weratna, Durhin Wong-Rieger, and Gustavo Scola. Now, the first case study that will be presented today is the development of a gene therapy by NRC's Cell and Gene Therapy Program and our collaborators. And in fact, the mission of our CGT program is to help accelerate the development of made in Canada cell and gene therapies for chronic diseases and for rare diseases. And ultimately, our objective is really to help make these therapies more accessible for Canadians. So to this end, we have established a Canada-wide ecosystem of over 60 collaborators to date. And these collaborators include SMEs and academics, hospitals, consortia and patients, government institutions, funding agencies, and international partners. And NRC has also allocated significant infrastructure investment to collaborators. And so together, we are happy to announce that this year, our first CAR-T cell therapy will be entering clinical trials. And we have also selected a lead candidate for a gene therapy for a rare lipid disorder that is prevalent in Quebec. We have also developed a series of tools and platforms that enable or significantly reduce the cost of CGT by manufacturing. Now, these tools and platforms, they include high throughput gene editing tools and an iPSC biobank for the development of off-the-shelf CGT products. They also include engineering cells for viral vector manufacture, targeted lipid nanoparticles for delivery of CGT, and solid tumor CAR-T therapies. So currently we are working towards transferring these technologies we have developed to national and international recipients. We are organizing a CGT bioasset transfer event at NRC for June 13th, so please stay tuned. Before we move on to the introductory talk, we would, however, like to mention that since the beginning, NRC's LPLD gene therapy development was led by our colleague, Dr. Danica Stanimirovic. Dana had accepted to present this project to you today. However, it is with great sadness that we have to inform you that a few weeks ago, Dana suddenly passed away. So Dana was a pioneer in NRC in gene therapy development for rare diseases. She also made an important contribution to our CGT program's collaboration with Health Canada on facilitating regulatory approval in Canada for novel gene therapies in the context of the Project Forward initiative. She also supported our collaboration with CIHR to create a novel grant opportunity for gene therapy development for rare diseases and for working towards its readiness for clinical trials. So we would like to underline Dana's tremendous impact on our CGT program, on our research center and our collaborators by dedicating today's symposium to her. And now I would like to welcome Dr. Christopher McMaster. Dr. McMaster is a scientific director of CIHR's Institute of Genetics and will be giving the symposium's introductory talk. So Dr. McMaster is a professor, and he's also the former head of pharmacology and director of cheminformatics drug discovery at Dalhousie University. He is a recognized leader in genetics, biochemistry, and cell biology, and his research encompasses a broad range of interests, including basic biochemistry, genomics, and ethical considerations in the field of human genomics. Dr. McMaster has identified a potential therapy for congenital sideroblastic anemia and is developing treatments for muscular dystrophy and familial exudative vitreoretinopathy. He received his bachelor's in microbiology and PhD in biochemistry from the University of Manitoba, followed by a postdoctoral fellowship at Duke University Medical Center. Dr. McMaster, the floor is yours.
Thank you very much. I will be getting my presentation shared in a second now, I hope. Just have to go into full screen mode. Does everything look good? Perfect. Alrighty, I was given the very simple task of trying to overview the Canadian rare disease landscape from a research perspective, which was not so simple. I will say it's a bit of a CIHR-centric view I'm going to give you at the beginning, and then do my best to try and expand out into what the rest of the country and other funders are working on. Alrighty, so one of the things Canada did not have that I think we needed to have to understand rare diseases in general, but diseases in general, was a pan-Canadian human genome library. One of the things we did in Canada every year was we sequenced 20 to 25,000 human genomes or exomes as part of research studies, never mind in the clinic. But these were all disparate and could not be captured under one umbrella. And we've done this for about 5-6 years now. You can imagine we'd have 150,000 exomes or genomes as part of NICE cohorts already done if we would have had a library with which to deposit these, which we did not. So the library is to bring all genome sequencing efforts in Canada under one roof so that researchers can have unprecedented and diverse genomic data for analysis. And it will also be tied to either cohorts or healthcare records so that folks can actually sort out genomes from phenomes and figure out what keeps us healthy, what predisposes us to disease, including rare diseases. This was recently funded by IG as well as a lot of other partners within CIHR. And it's already moving forward in terms of getting up and running. It's been funded for about 2 months now. The other thing that this library will facilitate that we could not do before was that we had no idea in Canada what our variome looked like. You know, most genomes are people of European descent. In Canada, was that the case or not? Now that we've got a library, we can ensure that when we're looking to translate discoveries from genomic research into the clinic, that we've actually got all of the reference genomes appropriately covered across to Canada so that nobody really gets left behind when it comes to diagnosis. To do so, one of the other problems that we had in Canada was there was no set core elements for genomic research consent. A lot of the consents were very limiting. So together, the Institute of Genetics, along with a lot of ethics and policy experts, put together core elements of participant consent for Canadian human genomics research. This is now sort of the go-to government for the Canadian Association of REBs that they look to see what needs to be in a consent for genomic research. It's also being used by everybody that's participating in the library, et cetera. But this enables data sharing. Most importantly, enables data for future use. Also, most importantly, it is disease agnostic. So many consents in the past would have a drop dead date as far as the genomics research goes, which means you could not use it past a certain date. It was also specific to a certain disease, which meant you could not then put it into, for something like example, the Canadian Human Genome Library, and it'd be useful. The data will be anonymized for research, but the material findings, if you come across a material finding, will enable recontact. And this is all being computerized, et cetera. Most recently, we announced and we'll be funding a national clinical trials and treatment network. This will be a first in Canada in rare diseases. So we'll develop a platform to support rare disease clinical trials for the profit and not-for-profit sectors. This will also maximize the impact of government and Canada's investment in regulatory modernization, as well as, you know, things like NRC's efforts and other folks' efforts to actually move medicines into the clinic and hopefully develop a hub to attract international clinical trials. One of the issues in Canada is that only 60% of rare disease medicines that are approved by the FDA and or the EMA are actually approved in Canada. There's two ways to go about this. One would be reciprocity between Health Canada and other agencies. And the second would be to actually bring clinical trials into the country, because if you already are participating into the clinical trial, most sponsors will then tend to try and register the projects where they've already got a relationship with the regulator through the clinical trial process. So one, this will hopefully get new medicines for rare diseases into Canadian, mostly kids, as soon as they're available, like during the clinical trial, which means they're free during the clinical trial. And with a relationship with the regulator as the clinical trial is going, will encourage sponsors to actually then apply in Canada to actually have these medicines available ASAP for Canadian folks. The second thing that's really annoying when it comes to rare disease kids is that they're not counted. If you look at pediatric hospital databases, if you look at administrative databases for provinces, everybody uses ICD codes to determine, you know, what you're doing. If you got pediatric cancer, then what kind of cancer, and on and on and on. For rare diseases, there's just a couple hundred for the 7,000 rare diseases. However, ORFA codes have been developed that have all the rare diseases in there, including a line for rare disease undiagnosed, which is really important because that means that you can now actually count these kids. So you can figure out what is the cost to the healthcare system, what's the hospital use, and on and on. What are the mortalities? What are the morbidities? What are the indirect costs to the healthcare system, patients? Retrospectively, when people do this, they find that rare diseases are by far the most costly, the biggest cost to any healthcare system in the pediatric setting, and that it's the major reason for mortality amongst kids in almost every country in Europe, North America, most of the global north. The second thing is with ORFA codes, you can automatically generate patient registries because you have a code for every kid that corresponds to their disease. And you can also work on the economics, because how should you know how much you should spend on care, diagnosis, treatment, and research if you actually have no idea what the burden to the healthcare system is? So this will enable that for the first time in Canada. Second thing, another thing is that rare diseases, now that genomics has become quite cheap, is that we still have genomics as sort of the test of last resort for many kids. On average, it's about 5 to 7 years to diagnose a rare disease in Canada. And that's because medical geneticists are usually the doctors of last resort. So you go through a bazillion specialists, bunch of tests that are unnecessary, and then you end up in medical genetics where a genome or an exome or a panel will be ordered. And that's too late in the process. So we've now put out a research call to say, okay, where within Canada should this exist? Should it be at birth? Should it be in the NICU? Should it be at first presentation? So that we can actually get the appropriate test, genomic test, to the right child at the earliest time point, because the hit rate now for genomic testing for rare diseases is 50% and increasing exponentially every year as more and more genomes are being sequenced. So this is another thing we're funding. Let's just try and get rid of the diagnostic odyssey as best as we can. And the other thing we're funding is a rare disease models and mechanisms network. And this is, when a child shows up with what is clearly a rare inherited disease, and you've got a candidate gene, a lot of times you need to model it in some sort of mouse or organoid or something else, model organism of your choice, to prove that you've actually got the right gene. So if the model organism ended up with a disease that looks like what the kid has when you make the same variant in the model organism, proves you've got the right gene. In Canada, we've had this up and running for over a decade now, and it's extraordinarily successful in ensuring that you actually have a causal gene for a rare disease. It's actually now been modeled in Japan and Australia and Europe and many other countries, where we basically gave them all the software and hardware and processes on how to do it. And it's now become international in its capacity to now validate clinical gene discoveries. And you also get a model to study the disease as part of the process, so scientists can then work on mechanism of disease and potential therapies at the same time. And finally, we're looking to bring, along with NRC, as was mentioned earlier, gene therapies to clinical trial readiness. So we're trying to maximize the Government of Canada's investment in biomanufacturing, in this case, AAV vectors. And so we're funding scientists to work with the NRC. One of the things we're not very good at in Canada is going from mouse to human. We're very good at treating and curing our mice, and we're very good at clinical trials at phase 2 and 3. We are not very good at moving from mouse to human. This is to encourage and fund folks to work with NRC, who has got a similar project for a rare disease, the lipoprotein lipase deficiency you'll hear about in a bit, to work on 2 or 3 other vectors, therapies that are using AAV vectors, NRC's expertise, to be able to get them for first-in-human clinical trial readiness. Another nice thing about working in the rare disease space, especially for kids, is if there's one kid in the States that has that rare disease, which is almost always the case, and the FDA also approves it, you can get a pediatric priority review voucher from the FDA, which is worth about 150 million Canadian dollars, to continue to accelerate your own gene discovery processes. And finally, we all know now that the biggest issue in interpreting which genes may or may not cause a rare disease is not the cost of genomes or the speed of genomes, it's actually the analysis of the genomes. So we're funding a nationwide bioinformatics and computational biology and health data science training platform. Any graduate student or postdoctoral fellow in any university will be able to plug into these courses, such that you can either use the plug-and-play software, so you can analyze your exome or your genome or your RNA-seq data, all the way through to people who want to find out, using AI and machine learning, better ways to analyze genomes and associated health records to figure out what keeps us healthy and what predisposes us to disease. The other thing we're looking at is patients. We wanted to determine what are the indirect healthcare costs to patients living with inherited diseases, identify the most notable indirect social costs that families are actually having to pay out of pocket, and then try and present to policymakers changes to policy that can then enable these folks to access appropriate supports, which right now many times they can't, especially when you're working on getting your child diagnosed, but they don't yet have a diagnosis. And so these projects are almost complete, and we're in the process of wrapping up the findings so we can present it to policymakers to hopefully move the needle with policymakers with respect to getting more supports for parents who are awaiting a diagnosis for the kid with a rare disease. Beyond CIHR, there are lots of other folks, and this is not everybody, but Genome Canada is looking at clinical genomes through All for One and GAP projects. They've got a rare disease cohort through Care for Rare. They're thinking of, there's cancer cohorts that are out there. And in addition, they're looking towards funding population cohorts, Indigenous peoples cohorts, which are already funding through silent genomes, as well as contributing all future cohorts to the genome library to expand it. NRC, of course, has and is looking to increase GMP manufacturing capacity for biological therapies, including gene therapies. And as mentioned, they are collaborating with us through academic collaborations to enable first-in-human clinical trials to try and bring gene therapies into the clinic, but also bring them in a cost-effective manner. Health Canada is direct funding for high-cost therapies that is in the progress of trying to hammer out agreements with the provinces at this point in time. They're also looking at genetic disease drug approval modernization as an internal working group, and they're also looking at the potential for reciprocity of approval with the FDA and the EMA. But these are all things that are just sort of policies that are being worked on and looking at viability of. The biggest announcement, I guess, in terms of dollars is just announced winter 2023, which was Canada First Research Excellence Fund, $125 million for One Child, Every Child. It connects all U15 universities as well as 132 organizations from 25 countries committed to essentially rare disease research that will hopefully, and they want to act as the umbrella organization to accomplish what we hope will become a learning health system. Right now, we have everything in Canada required to do this. We're starting to move towards prompt patient diagnosis and care through the rare disease models and mechanisms process, the improved diagnosis to get rid of the genetic, the diagnostic odyssey, the genome library, cohorts, and bioinformatics training. We're accelerating development of advanced therapies. The national consent is helping. The AAV gene therapies, the GMP manufacturing at NRC, and the shared research projects with CIHR is starting to move that needle. And then the clinical trials and monitoring, well, we just set up the clinical trials and treatment network. We're bringing in ORFA code so we can count and find all these kids, and Health Canada is looking at regulatory modernization, followed by accessibility of treatment in terms of looking at the socioeconomic burden of kids and families with rare diseases to determine, accessibility to treatments as well as accessibility to diagnosis and any issues with that. As well as potential regulatory reciprocity between regulators. As you can see, we have everything required, but it's dreadfully fragmented right now. There's Care for Rare, there's All for One, there's HostSeq, there's silent genomes, rare disease models and mechanisms, there's MyCern, there's CORD, there's One Child, Every Child. We have all the pieces to have a nice learning healthcare system where all of this could potentially connect. What we don't have is sort of a mothership that is actually acting as a process by which all of the research and all of the clinic can be connected in a learning health system, so that as we learn more and more about genomes, it keeps us healthy and predisposes us to disease, and in this case with a focus on rare disease and the therapies that are coming down the pipeline. It is everybody's got a piece of the pie, and we can connect all the dots. Hopefully One Child, Every Child, and this is what they're hoping to do along with MyCern, and of course CORD's always looking to do this as well, is act as a bit of a mothership to coordinate all of these activities under one umbrella so that it's a much more seamless than the sort of hit and miss processes that are, it's not hit and miss, but the sort of what looks like discordant, but when you put them in a nice pretty diagram, we do have all the pieces to actually be able to do this. We just need a mothership organization to be able to take the time to do all the coordinating. And again, reminder, Rare Disease Day on the Rare Day of February 29th, hence why it's Rare Disease Day, and thank you for your time. I will attempt to stop sharing.
Thank you very much, Dr. McMaster. Our first case study today will feature Dr. Maria Moreno. At the end of each session or after each speaker, we will have some time for a couple of questions from the audience. So we invite you to put your questions in the Q&A box, which you will find at the bottom of your screen. So Dr. Maria Moreno is the head of systems pharmacology section at the NRC, where she has developed a preclinical facility that integrates pharmacokinetic, drug biodistribution, and efficacy studies. She is also a technology thrust lead in the multifunctional antibody-based therapeutics program and is leading the development of antibody drug conjugates for cancer treatment. Dr. Moreno investigates innovative drug targets and delivery modalities and has established partnerships with various SMEs. She has published over 60 scientific papers and has 8 patented innovations. Now, Dr. Maria Moreno's presentation is entitled Canadian Path, a Gene Therapy for Lipoprotein Lipase Deficiency from Design to Clinic. And with that, I'll hand it over to you, Dr. Moreno.
Thank you, Freya. Good afternoon, everybody, and thank you for giving me the opportunity to be today here. Is very sadly, I'm actually here in the place of Dr. Danica Stanimirovic. Freya has already explained the very terrible circumstances for her absence, and I'm going to do my best to present this first case study that was really led by Danica Stanimirovic. She brought a very strong leadership, and not only the leadership in bringing one product to developing a product, a gene therapy product to make it accessible to Canadians, but also really establishing a very strong foundation for future gene therapies here in Canada. So I'm going to see if I can put my presentation here. Do you see the presentation?
Yes, perfect. Thank you.
Okay. So the title is Canadian Path Gene Therapy for Lipoprotein Lipase Deficiency from Design to Clinic. So I'm going to just start giving you a very brief overview of the disease we are targeting. So this is the lipoprotein lipase deficiency, LPLD. This is a monogenic autosomal recessive disorder. It's a rare disease that affects 1 to 2 in 500,000 of the general population. However, in several regions, the prevalence is much higher. And for instance, here in Canada, it is 200-fold more frequent in the Saguenay-Lac-Saint-Jean region. So we can consider this really as a very important Canadian disease. There are 2 variants, the P207L and the G188E, that account for 75 to 95% of the mutant alleles in the French Canadian population. With LPLD. And these mutations, sorry, I have a problem here. I have to resolve this because if not, I can't continue. For some reason I have, yeah, sorry here, yes. Maybe I can eliminate this. Yes. Okay, sorry for that. Okay, so there are 2 variants that really account for 75 to 95% of the mutant alleles in the French Canadian population with LPLD. And these mutations produce a complete lack of functionally active LPL protein. The result of this is, as you can see here, is that these patients exhibit plasma with this milky aspect, this milky appearance. And this is because the accumulation, very strong accumulation of lipid nanoparticles. And if you look at the right side, these patients also develop a very they have other presentations such as xanthomas. These are eruptive xanthomas. These are lipid accumulations under the skin. And the most acute manifestation and the most severe manifestation is acute pancreatitis that in some cases induce necrosis and can be life-threatening. So there was a therapy for these patients. The name of this therapy was Glybera, and many of you must have heard about this therapy because made the news for being one of the most expensive drugs in the world. But I would like to give you a little bit of the history of this drug. So just bringing you back, the genetic mutation that caused the disease was discovered by Dr. Michael Hayden and Dr. Colin Ross here in Canada at the University of British Columbia. And they also developed a gene therapy treatment for the disease. Unicure, which is a Dutch company, acquired the rights to develop and biomanufacture the therapy, and this therapy was branded as Glybera. They went through a very rigorous clinical trial and a regulatory process. And pivotal clinical trials were actually conducted in Canada, here in Quebec, by Dr. Daniel Goudet. The gene therapy was finally approved in Europe in 2012, but with a price tag of 1 million euros. So this is 1 million euros for just one treatment. So of course, there was no uptake of the treatment, and CISI, which was the Italian company that acquired the rights for commercialization in Europe, abandoned the commercial rights in 2017. However, long-term monitoring from the patients that were enrolled in the clinical trials demonstrated drug efficacy and lower burden of the disease. However, Glybera was never approved in Canada. So in 2019, there was an opportunity. There was a very interesting funding opportunity coming from the federal government that launched what was called the challenge programs here at NRC. And one of the first challenge programs approved was the disruptive technology. Again, I cannot see the top, for cell and gene therapy. And this challenge program has the aim of building a value chain model for engineered cell and gene therapies from discovery to commercialization. This program also had funding envelope to establish collaborative partnership as well as infrastructure management with key stakeholders that with the idea to bring these disruptive solutions to the design, development, and delivery of cell and gene therapy to patients in Canada. So with this in mind, and considering that LPLD was a Canadian disease, that there was a drug that unfortunately could not penetrate the market here or be actually accessible to Canadian patients. And with this program in mind, Dr. Stanimirovic decided to launch the first project in this program that aim at developing an improved LPLD gene therapy and make it available to Canadians. So here is a schematic that present the overall objectives and partners of this program and this project. It was launched in 2019, and NRC undertook the challenge of designing an improved LPLD gene therapy, also established a scale-up and purification process, as well as established analytics with the idea of the process being transferred to a Canadian CMO. The University of British Columbia also received Dr. Hayden's lab, also received funding to perform the preclinical evaluation of these novel therapies. And there was NRC had infrastructure investment, and through an RFA, the Ottawa Hospital Research Institute was selected as the Canadian viral vector CMO that will be producing the GMP material for clinical trials. Later in the project, we engage Ecogene 21 with Dr. Godet, who was the pioneer and the clinician that ran the clinical trials for Glivera. And they are now going to sponsor the clinical trials. They are involved in the toxicology, in the CT application, and the clinical trials hopefully will be happening in 2026 in Quebec. So as you can see, the project went beyond just simply bringing a gene therapy product to the Canadian patients. It also established a value chain, Canadian value chain, and a novel model of collaboration to really develop new gene therapies in the future. So these are the key collaborators that have participated in this project. As I mentioned, Danica Stanimirovic was the leader of the project in collaboration with Dr. Michael Hayden from UBC. We had the fortune to work with Karim Kugas, our project manager, that has really been key for coordinating and maintaining all the communication between the different parties. And these are the different modules that have been part of this process and the leads. So we'll go one by one just so that everybody feels that has been part of this project is indicated. So we have for design and selection from NRC, the viral vectors team, and from UBC, Dr. Colin Ross. From preclinical evaluation, UBC, Dr. Colin Ross and Dr. Neil Mehta. And from NRC, the systems pharmacology team. From the bioprocess development and scale up, from NRC, the upstream, downstream analytics team. For bioprocess transfer to CMO, from NRC, we had the upstream, downstream, and analytics team. And from OHRI, Dr. John Bell. And for the CT and clinical trial, we have from Ecogene, Dr. Godet, and from UBC, Dr. Michael Hayden. So going a little bit on the science, so this is the initial vector, the Glivera vector, and these are some of the genome elements that are present in the vector. It's an AAV1 serotype, and it had a CMV promoter, a very strong promoter. The transgene was the LPL naturally occurring gene variant, the S447X. This is the variant that was tested for Glivera and was the most effective for gene therapy. And then it contains the WPRE, the Woodchuck hepatitis virus post-transcriptional regulatory element that was included to improve a viral titer. So when NRC started on the design of the new constructs and the new AAVs, there were some objectives that we wanted really to tackle. One was improve efficacy and potency, so to reduce the cost of the gene therapy treatment by potentially reducing the dose. So for this, we had to choose an AAV serotype with best transduction capacity of human skeletal muscle cells, choose a very strong promoter with a good expression in the muscle, and design a codon-optimized version of the LPL sequence. We really wanted to reduce immunogenicity, and for that, we decided to choose AAV serotype with low antiviral titers or with low exposure, and reduce the CpG content that has been shown to trigger innate immune response. And also, we wanted to reduce the potential long-term safety liabilities. And for that, we look at eliminating the WPRE element that has shown to have some oncogenic potential, although we didn't see any of these in the Glivera construct. So all these new designs were benchmarked against the original Glivera. Just to give you a sense of the scope of this project, we produced over 70 constructs. We tested 6 different promoters, 2 different reporter genes for biodistribution studies. We look at 5 different LPL sequences. We tested the elimination of WPRE, and we also tested 3 different polyase sequences. So just a little bit of the data that was generated for biodistribution studies. This was how we started looking at the tropism of different serotype and promoter combinations. In this case, we look at AV1 serotype and the AV8 serotype, AV8 being with less exposure and with less pre-existing antibodies in the general population. And then we look at 3 different promoters, the CMB that was present in Glybera, the CAC promoter that is a very strong promoter, constitutive promoter, and then the delta use promoter. This is a skeletal muscle specific promoter with the idea that it would maybe improve the potency. And then we substitute all the LPL transgene by luciferase. By doing this, we were able to monitor the biodistribution of the different constructs in vivo in longitudinal studies over time. We use various different routes of administration: intramuscular, intravenous, subcutaneous, and fat-pad, and 3 doses were tested from 10 to 11 to 10 to 13 genome copies per kilogram. And animals were imaged over time and using and looking at bioluminescence imaging. So you can have here just a snapshot of one of the routes of administrations: intramuscular, dorsal and ventral images. And here from these studies, really what we wanted to look is at the tropism of these different constructs. And what we found basically is that for all the vectors that have either the CMB or the CAC promoter, there was a very strong luciferase expression in the injected skeletal muscle sites. And this expression was much lower when we were using the skeletal muscle specific promoter, the delta use promoter. So it was 5 to 10-fold lower. And another very interesting aspect or feature is that the AV8 CAC promoter was able to not only transduce the muscle, but also the liver. So then we moved to do the efficacy studies. This was done at UBC by Dr. Neil Mehta. And here what we had is we compare again the 2 serotypes, AV1 and AV8, 3 different promoters. And we had the LPL codon and non-codon optimized. We look at 2 routes of administration, intramuscular or intravenous, and 2 different doses. And this was done in LPL knockout mice. And here you can see again the plasma of the different of the mice and different injected with different constructs. So you can see that in the positive in the wild type mice, the plasma is very transparent, but in the knockout mice is really milky. And with different treatments, we were able to reverse that milky look of the plasma, so clearing up and creating, reducing the triglycerides. This was the most important aspect. And we can see that this construct AV8 PRV59, which has the CAC promoter, was the one that had the strongest and the best to clear the plasma. It was also the one that produced the highest reduction in triglycerides in plasma. And really with this construct was more effective, at least 10-fold. It was effective at 10-fold lower doses compared to Glybera. So this was our lead at that moment. So just as a summary, the efficacy with a single dose of this construct, AV8-PRV59, we were able to correct LPLD at 10 to 100-fold lower dose compared to Glybera in mice. In terms of biodistribution, we saw that there was gene transfer to both liver and skeletal muscle. In terms of toxicity, we didn't see any toxicity following intramuscular delivery. No antibodies against the transgene were detected before or after administration. And just again, the schematic of our lead. Now, there were some other aspects that we wanted to tackle. One was to remove some potential oncogenic liabilities, and this was the idea of removing the WPRE element. It was included as because it increases the transgene expression for a variety of viral vectors. However, it's not a critical component, and there is always a potential oncogenic risk, although we think it's very low because it was tested with Glybera. However, this element also has 37 CpGs, so eliminating it could just also support or remove the CpGs that were also something we wanted to do. So the question is whether by removing it, we were losing efficacy or potency of this, the construct. And here, just to show you that no, the removal of WPRE did not affect the potency of the vector. So that was good news. The next step was to remove CpGs from the promoter and the open reading frame of the transgene. And the question is, could this affect efficacy? So we produced several constructs with different CpG levels, in some cases removing up to 77% of the CpGs. And unfortunately, we noticed that there was a very significant reduction in the potency of these constructs. So we decided to go back, and instead of removing the CpGs, to pay attention and focus only on immunostimulatory CpGs and convert them into neutral CpGs. So this was the last step we did. And we created a couple of constructs, and we found that there was one of them really performed very well, no difference to the original AV construct, and that with the same potency as the original. So right now, this is really our lead. Is an AV8 serotype with a CAG promoter. We have reduced the immunostimulatory CpGs to just, we only have now 20. And we are now moving forward to just complete the process of upstream and downstream. So this is our project plan from here to the clinic. So we are at the stage in which we are improving the process and transferring the process to OHRI. OHRI is going to produce a GMP engineering run of 50 liters. With this, we will be able to run the GLP toxicology studies. This GLP toxicology study has been already endorsed by Health Canada, with which we had a meeting a few months ago. And with the results of these studies, we will be submitting the CTE. There will be GMP clinical material production of 200 liters for our clinical trials. And we are really hoping to start these studies, these clinical trials, in the first half of 2026. So with this, I just want to say that really, we are building here the foundation for accessible gene therapies in Canada. With this project, it started simply with building on past experience with the first Canadian approved gene therapy. Then we have been able to establish a viable clinical scale biomanufacturing capacity for gene therapies in Canada. We are engaging advocacy groups to support new models and access to treatments. We are engaging federal and provincial governments and Health Canada to innovate in clinical trial design and delivery. And with this, I think we are creating really a lasting footprint for future gene therapies in Canada. And I just want to thank the team. There were many people involved. As you can see here, these are just the names of the teams that participated. I want to thank especially UBC for the preclinical studies, OHRI, Ecogene 21, and BC Cancer, Virica, and University of Alberta that have also participated in other parts of the project. And with this, I'm ready for any potential question.
Thank you so much, Maria.
Thank you.
In the interest of time, we're proposing to have the question and answers a little bit after the last presentation. And so, John, I'll hand it over to you now.
Okay, fantastic. Thank you. And again, thank you, Maria. So I'd like to welcome our second case study speaker, Dr. James Dowling. Dr. Dowling is a clinical scientist at the Hospital for Sick Children and a professor of pediatrics and molecular genetics at the University of Toronto. His expertise is in childhood neuromuscular disorders, and his research focuses on gene discovery and therapy development for congenital myopathies. He is also a member of the executive board of Treat NMD and the World Muscle Society. Dr. Dowling received his BSc and MSc from Yale University and his MD PhD from the University of Chicago. He did his residency in child neurology at the Children's Hospital of Philadelphia and completed postdoctoral research at the University of Pennsylvania and the University of Michigan. You can see his full bio in the booklet that's circulated through the chat. And I'd like to actually welcome Dr. Dowling, who his presentation will be entitled Genetic Therapies for Rare Neurogenic Disorders, the Sick Kids Experience. With that, I'll hand it over to you, Jim. Thank you.
Thanks, John. Let's see, hopefully my screen. Okay, is that visible?
I can see that.
Excellent. Thank you, and thanks for the invitation to speak today. I'm going to provide an overview of the gene therapy experience that we've now been developing at Sick Kids with a focus on neurogenic disease, even though certainly our experience has not been limited to that disease arena. So I just have a few disclosures to present. The most relevant for today is that I'm going to be talking a little bit about some sponsored research that was from funding provided by Astellas Gene Therapy for looking at preclinical research in X-linked myotubular myopathy. These first slides are really, fortunately, already been covered a bit by the previous speakers, but just to echo a little bit of what Chris was saying, rare disease, which is defined as affecting less than 1 in 2,000 individuals, is really not a rare problem. There's estimated to be about 10,000 rare diseases and affecting more than 1 million Canadians. And these are by and large genetic diseases. And as Chris emphasized, these carry with them incredible health needs and health burden and are amongst the severest diseases that we encounter. And I think one fact just kind of underscores this, which is that 30% of children with rare disease will not make it to their 5th birthday. There's obviously therefore a critical unmet need to identify and implement therapies for rare disease. And to start with kind of the sobering fact, which is that it's estimated that right now there are identifiable treatments for less than 5% of all of these rare diseases. But as Chris also emphasized, and I think you were able to get a flavor from the last talk, we now really have the tools to create and develop precise genetic therapies for most, if not all, of our rare disease patients. And I think the charge to all of us now is how we get this done. Just a reminder that I'm going to be talking about gene therapy, but it's only one of several different genetic therapy modalities that really span the entire molecular genetic central dogma. So therapies that could be targeted at DNA, that could be targeted at RNA, either as RNA therapies or therapies that modulate a transcript, and then protein-based therapies. And again, these can be either protein therapy itself, such as enzyme replacement, or else therapies that help a mutant protein function better, such as a chaperone therapy. But again, I'll be focusing specifically on DNA-based gene therapy or gene replacement therapy today. And I think you got a good overview from the previous talk, and in particular, some of the key elements related to gene therapy. I just thought I would throw up the Health Canada definition, so we are all talking about the same thing, which is an experimental approach where the faulty gene is fixed, replaced, or supplemented with a healthy gene so that it can function normally. This is really the definition of gene replacement therapy. And all gene therapy operates under a pretty basic structure of a transgene that has a promoter, the gene itself, and then some additional regulatory sequences that's packaged into a delivery construct. And in most cases now, that's a viral vector. And as was pointed out in the previous talk, AAV is the most common viral vector used for systemic and or regional gene therapy in humans. Importantly, gene therapy is quite an iterative technology in that really can plug and play with the different AAVs and a whole host of genes. And this is really suitable for a large number of diseases and in novel approaches such as platform or basket development. And that's particularly true because most of the toxicity associated with gene therapy really relates to the AAV itself. And so therefore you can get some predictions of class-related toxicity to help with ease of development and clinical planning. So again, I'm focusing on our experience with gene therapy in the neurogenetic sphere at SickKids, but it's just to say that there's additional ongoing work, particularly related to ocular gene therapy at SickKids. And I'm going to divide my talk really into three spheres. One is some of the work we're doing in the clinical trial arena related to gene therapy. And there I'm going to focus on our program related to X-linked myotubular myopathy. I'm going to talk a bit about clinical treatment, and I think I'm going to talk about the SMA gene therapy, which is really, to me, if not the biggest success, certainly one of the biggest successes in Canada of gene therapy implementation. And then I'm going to talk about how this has led to our consideration of a gene therapy committee to really help oversee both clinical trials and clinical treatment. So to start, I want to introduce X-linked myotubular myopathy. So this is an ultra-rare disorder affecting 1 in 50,000 boys, caused by loss of function or expression mutations in the X-linked MTM1 gene. This is a very severe condition associated with 25% mortality in the first year of life, and really nearly complete mortality before patients reach adulthood. Those that do survive that first year of life have significant comorbidities, including 80% requiring feeding tube, ventilator support, and wheelchair support. Currently, there are no treatments or disease-modifying therapies, and this is a disease very well suited for gene replacement therapy because the gene itself, MTM1, is small and fits within pretty much all viral vectors. So a gene therapy program was launched several years ago. This grew out of a collaboration between 2 American institutions and Genethon in France. And it's an AAV8 based gene replacement therapy. And this therapy was tested in a mouse model as well as in a spontaneously occurring dog model of the disease. And in both models showed great efficacy and really very little safety concern. So there was a lot of enthusiasm based on this preclinical data to bring gene therapy to patients with XLMTM. And that resulted in an industry-sponsored clinical trial called Aspiro, which is a gene replacement therapy trial for XLMTM. And this trial, again, grew out of that preclinical data as well as several natural history studies that were conducted both academically and industry-sponsored. It was a delayed treatment trial, so one arm of the patients got the therapy immediately and others were randomized to a 1-year delay as the comparator group, so there was no true placebo. And the primary efficacy was twofold. One, there was a a motor outcome measure called the CHOP and TEND. And the second outcome measure is actually something that we developed at Sick Kids, which is monitoring the time on ventilation, which is a pretty simple but robust measure of respiratory burden. And obviously the co-primary was safety, and was carefully monitored throughout the study. As I'll mention in a few slides, the data from the study has now been published, but I'm just going to highlight the really quite amazing efficacy of the study. So to date, 24 boys have been dosed with AAV MTM1, and it's promoted quite remarkable clinical improvement, which I'm slowing here in two different means. One is on the left, that motor function scale that I mentioned to you called the CHOP and TEND. This is a composite score that really highlights motor function across multiple domains. And you can see here in gray the typical untreated MTM1 patients. So this is the delayed treatment group as well as the natural history cohort, really with very low CHOP and TEND scores and no change in the score over 48 weeks. Up here is the clinical trial cohort, and hopefully you can appreciate the dramatic increase in the CHOP and TEND score. And actually, all of the first several patients treated reached the ceiling of this score, indicating a relatively normal return to motor function. I think perhaps more dramatically is the figure on the right, which shows the hours of ventilatory support of each of the individuals in the study. And this is the first 12 patients in the study, but we now have the data for 24 individuals. The gray box again is the untreated cohorts, and you can see here no change in ventilatory support over the 84 weeks of the study. There's actually two control patients here, the other one here in gray. And remarkably, the first 12 patients all went from at least 12 hours of ventilatory support a day to full ventilatory freedom. And I can say in particular, the 5 patients that we've treated at Sick Kids have all achieved full ventilatory freedom and in fact have had their tracheostomy removed, and are now breathing independently without any difficulties. I'll go very briefly over this just to show that the therapy did not only improve function, but also improve the muscle structure. Actually, it was near reversal of all the pathology seen at baseline. And again, these data have been now published in 2 papers, one in Lancet Neurology and the other in Biomedicine. Unfortunately, there's a big but associated with this amazing efficacy results. And that's that in the study, very surprisingly, 4 of the 24 individuals developed severe liver toxicity leading to their death. And several others also developed some liver dysfunction as well. This was a completely unexpected SAE and not something that's been seen in any previous gene therapy trial. Even though there has been liver toxicity noted, this high percentage of severe disease that's resulted in death was unprecedented. And one thing to note is that while the AAV, as mentioned in the previous talk, will go, the AAV8 goes both to the muscle and the liver, the gene was driven by a muscle-specific promoter. So expression was only found in the muscle. So it wasn't due to toxicity, toxic increase of MTM1 expression, but rather to the introduction of AAV to the liver. And unfortunately, now the trial is on clinical hold due to this unexpected result. So where are we now with, or what do we know about liver disease in XLMTM? So historically, this was a pure muscle disease, despite the fact that the MTM1 gene is ubiquitously expressed. But as we start to comb over some of the newer natural history data, we're starting to get a flavor that there actually may be a muscle or a liver disease associated with XLMTM. So we completed a natural history lead-in study to the gene therapy trial and found that 90% of the patients had abnormal LFTs, liver function studies, suggestive of some liver injury. And there's now subsequently been a publication shown here of a small case series of patients who had full-blown cholestatic liver disease related to the disorder, suggesting perhaps that XLMTM may actually be a liver disease in addition to being a muscle disease. So we wanted to understand this further and decided to return to the model organisms that we used to study XLMTM, which again previously did not show any liver dysfunction. However, when we published our zebrafish model of MTM several years ago, we did note some abnormalities in the liver. So we started there as a way to try to understand whether or not MTM1 loss by itself could result in liver disease. And sure enough, in a study we just recently published, we were able to convincingly demonstrate that loss of MTM1 results in a cholestatic liver disease in the fish, very similar to what we're now seeing in patients. And that this is a liver autonomous defect because we can rescue it by re-expressing MTM1 in the liver. Of course, we really want to know what's happening in mammalian models, because that will certainly be our springboard to better understanding how we can understand the interplay of AAV and potentially develop therapies. And so we've now gone on to work in the mouse model of the disease and have been able to uncover liver phenotype that was previously not observed in the mice. And furthermore, we've been able to show that we can demonstrate this significant exacerbation of liver disease by introducing AAV into the mice. So really, we've been able to recapitulate all of these features that we've been seeing in the patients, and that hopefully this will provide us with a springboard for being able to understand why liver disease occurs in MTM, why it's variable between patients, what's causing this this sort of two-hit model with AAV, and how we might be able to treat it. And that's work to be continued, but that's where things are with the XLMTM gene therapy program. So I think this is a nice example of how we were involved with a clinical trial study that also led to some important preclinical research that's now ongoing. So now I'm going to shift gears and talk about a clinical implementation example at Sick Kids, and that's a disease that probably many of you in the audience are familiar with. It's called spinal muscular atrophy. So SMA is a neuromuscular disorder caused by biallelic mutations in the SMN1 gene. It presents with several different clinical subtypes that are largely dictated by a modifier gene called SMN2. The most severe subtypes, type 1 and type 2, again, are quite severe, with type 1 patients in the untreated setting typically dying by age 2 years of age. So there was quite a high medical burden and unmet need for therapy. But excitingly, now multiple therapies have been developed for SMA, including ones that modulate SMN2 levels and SMN1 replacement, gene replacement in a medicine called Zolgensma. So Zolgensma is an AAV9-based SMN1 delivered systemically via IV infusion. There are now abundant clinical trial results showing its efficacy in both pre-symptomatic individuals as well as early symptomatic infants less than 6 months of age for SMA type 1 and 2. And so these are patients with 2 or 3 copies of the SMN2 gene. Again, it showed a very strong efficacy signal in terms of improved motor milestones, freedom of ventilatory support, and prolonged survival, and is also generally well tolerated, with side effects that have been seen really not only in this clinical setting, but really with all gene therapy. This was a drug that was Health Canada approved and now has a provincial funding envelope in Ontario for patients with SMA and 1 to 3 copies of SMN2 who are under 180 days of age. And to note, at the time of approval, Zolgensma was considered the world's most expensive drug, with an approximate cost of $2.4 million Canadian. It's since been passed by several other gene therapies, which are not available in Canada, but are certainly worth discussing, perhaps at our discussion stage. So this is our process for the approved gene therapy. And the other innovation that's occurred with this is the newborn screening process. So since these therapies have come on board, newborn screening for SMA has been approved in Ontario and now in really, I think, pretty much every province in Canada. So we're detecting patients within 7 to 10 days of life based on newborn screen, and then having them seen in our neuromuscular clinic within 24 hours, where we then get consent for moving forward with AAV9 gene therapy if the family is interested. And we're typically administering gene therapy within 3 weeks of life. And that's a stage that's pre-symptomatic for patients. And this is really the monitoring process. So we start immunosuppression 24 hours before gene therapy, continue it for 1 month, and then wean, and then monitor the patients really monthly, at least, continuously thereafter. So we've seen a very remarkable efficacy with this program that's quite similar to what was reported in the clinical trials. So many of our patients that have been treated are now walking independently and doing stairs, which is something that would never be seen in an untreated SMA1 or SMA2 patient. And we've had a very good safety signal as well, with only a few adverse events, which I'll mention in the next slide. So this has been our experience. We've effectively implemented newborn screening as well as pre-symptomatic treatment with either Zolgensma or the other SMN modulating therapies. We've successfully treated 13 patients thus far, which we think might be the most in Canada, but certainly is a good representative number to say that we're able to do this process very effectively. We've seen really dramatic effectiveness with the therapy. And we've also seen that most, if not all, patients experience some element of side effects. Typically, these are restricted to laboratory abnormalities. So we've had one serious AE related to thrombotic microangiopathy. And it should be noted that worldwide, there have been several reports of severe adverse events, including at least one individual who has died as a result of receiving gene therapy. All of these side effects, as well as the complex nature of both ordering and implementing the gene therapy, has led us to the conclusion that it really requires a specialized care team. And our experience both with SMA gene therapy as well as our clinical trials has led us to establish an in vivo gene therapy committee. So this committee is led by Dr. Hernan Gonarowski and a very talented nurse practitioner, Elisa Nigro. And this is the full group, and it represents clinicians and allied health individuals across multiple specialties, with the goal of providing optimized clinical care for patients with gene therapy, and really to centralize our care model for patients who do receive gene therapy, whether it's in a clinical trial or in a clinical setting, as well as to provide core support really across the hospital for anyone thinking about gene therapy related projects. So right now, our deliverables have been successful implementation of SMA gene therapy. We've organized and participated in 3 ongoing clinical trials, and we have, I think, 2 others that are planned. And we're developing standard operating procedures for different care areas related to gene therapy that are dealing primarily with the adverse events, but also with the ascertainment process. And we're hoping to publish these and disseminate these broadly, not just for investigators, the sick kids, but hopefully to benefit individuals, teams across the country. So just for the last part, a little summary and to reflect on experience so far and what I think the future directions are going. So from our experience thus far, we think that gene therapy is an incredibly exciting and effective modality that's potentially applicable across a broad range of patients, but recognize that it needs a specialized infrastructure and clinical team, both for clinical trials as well as for clinical care. And that there's an interesting and significant overlap between a trial and the actual clinical care in terms of, for a lifelong single dose therapy, they very quickly transition from a clinical trial to really having received a clinical product and requiring more standard and complex clinical care. As Chris mentioned, there's several industry-sponsored gene therapy clinical trials that are ongoing, and unfortunately, only a few of these are represented by Canadian sites. And so I think a big question is how we can attract more clinical trials to Canada. And perhaps this our new rare disease network may be a perfect opportunity to develop within it a gene therapy specific trial network. Chris also mentioned that there's several approved gene therapies, either at the FDA or in EMA. And very few of these are available in Canada. So far, 4 approved gene therapies in Canada, and several outside of Canada. And in talking to the companies, none of them really have concrete plans of coming to Canada at present. They have very vague plans, and that's certainly something that needs to be addressed. And one note related to that is that industry-developed gene therapy is extremely expensive. And so obviously puts quite a burden on the healthcare system, and is a key pivot point in terms of thinking about how we develop gene therapy. And then I think an important thing to keep in mind is that I mentioned this is an iterative technology. There's probably more than 1,000 rare diseases that could be treated right now with gene therapy, where we have the knowledge of the disease and we have the ability to make a suitable vector to treat it. One of the beauties of gene therapy for recessive disease in particular is that it's quite simplistic. If you've lost the expression of a gene and you can re-express it, that should really be a very viable therapy for most diseases. Many of these rare diseases are not commercially viable, and even if they are, the cost from pharma is exorbitant. And so the question is, how do we develop a pathway or think about a pathway where we overcome these barriers to make gene therapy for all of these ultra rare diseases where it's applicable? And I'm going to talk about one example that perhaps could be a springboard for future development. And this is a disease called SPG50, which is an autosomal recessive disease caused by biallelic pathogenic variants in the AP4M1 gene. It's a progressive condition that first starts in infancy, that then results in progressive loss of cognitive skills and motor function. And by the second decade results in severe spastic quadriparesis and intellectual disability and early death. It's an ultra rare disease, and there's estimated to be approximately 60 to 70 patients worldwide. Oh, sorry, my cat just fell on the on the computer. And there's only one patient in Canada. Since later today, as part of our panel, the parent of this child, Michael, will be on our panel. He'll be able to also answer some questions related to this. And so how do we think about gene therapy for SPG50? As I mentioned, the genetic therapies have multiple potential modalities, but SPG50 is really perfectly suited for gene therapy because it's a small gene that's progressive with a hopefully large window of therapeutic intervention opportunity. And based really on the amazing work of Michael's father, Terry, and Michael's family, they started the Cure SPG50 Foundation, which then nucleated this multinational group of academic and industry collaborators to develop a first-in-class gene therapy for SPG50. Not only did they nucleate this experience, but they also raised pivotal funding to support all of the phases of the development of this gene therapy. This is the path of SPG50 gene therapy. And so Michael was diagnosed in April of 2019. And at that time, there was really no concept of doing gene therapy for SPG50, no animal models, no cell models. And in this time span from 2019 to 2022, all of these things were completed. Animal models developed, AAV-based gene therapy developed and tested. Health Canada notice to move obtained, and less than 3 years after Michael's diagnosis, we were able to dose him in an N of 1 clinical trial at sick kids with SPG50 gene therapy. This is just the publication of the preclinical data. I wanted to highlight Stephen Gray, who was the scientist who developed it. This is treatment day, March 2022. This is me holding the gene therapy and hopefully not dropping it. And this is Michael with his dad, Terry, getting ready to receive gene therapy. And this is all of us with Michael getting ready to hopefully administer a successful and safe gene therapy to him. So what's happened thus far? So we're now almost 2 years into the clinical trial. And fortunately, the medicine has been very well tolerated with no grade 4 or 5 SAEs. That's not to say that there hasn't been side effects from the treatment and also from the immunomodulation related to it. This has resulted in careful ongoing monitoring throughout the whole 2-year process. Importantly and excitingly, we're starting to see some suggestion of an efficacy signal related to the therapy. And in particular, Michael's parents have noted improvement in his development and communication abilities, as well as some reduction in his spasticity. So to summarize, we performed a first-in-human single-patient gene therapy trial for SPG50. And we think this represents really one of the first N of 1 gene therapy trials for rare disease. Again, the trial is ongoing, but so far has been well tolerated with qualitative and now quantitative improvements in function noted. Based on these data, there's now a phase 1-2-3 trial for SPG50 ongoing in the US, and the first patients have been dosed. And I think, of course, the big question is this just a one-time exceptional thing, or is this something that we could use as a springboard for developing small N gene therapies and genetic therapies for ultra-rare disease? So at Sick Kids, we decided that this is something that required tackling with a real hospital-wide systematic approach. And really our goal is to shift the current paradigm of therapy, which really to me is quite reactive. We diagnose a patient with the disease and then we say, are there therapies out there that already exist that could be helpful? If not, well, we kind of sit back and wait for them. And can we shift to a paradigm where instead we're proactively developing the therapies, where after we diagnose the patient, we say, what could we do? And let's go ahead and do it. And that's the goal of our advanced therapeutics node at Sick Kids, really to consider all the different potential modalities for genetic therapy to apply for rare genetic disease. This has been codified into a virtual center that we're calling our Precision Child Health Center for Advanced Therapeutics, or CRAFT. And really the goal of CRAFT is to go from diagnosis all the way to personalized therapy implementation. One of the key principles that we want to focus on is the idea of therapy that's both equitable and inclusive, and that's not restricted just to specific patients that have the opportunity because of access or means to have a therapy, but rather for all of our patients, and to base this on need and suitability for therapy. And to help address this, we've formed an Advanced Therapeutics Advisory Board with representation from really several key groups, including bioethics, our research ethics board, patient advisors, as well as clinicians and scientists. This is our board, and I think we've got really excellent representation across the hospital to hopefully make this process one that is available ultimately for all of our patients with rare disease. So just to summarize, I'd say that gene therapy is an incredibly exciting modality with potential to treat hundreds, if not thousands, of rare disease by addressing their root cause. And we've been fortunate to be participants in some successful gene therapy clinical trials, as well as one of the first available gene therapies in Canada for spinal muscular atrophy. There's important and unique clinical care requirements for gene therapy. And these obviously need to be considered as hopefully more and more therapies come on board. And also gene therapy has a high cost, which clearly has to be addressed. We were able to show in proof of concept that it can be developed for even ultra rare disease, and in particular as an individualized gene therapy for one specific patient. And as I mentioned, we have the ability to make gene therapy for hundreds, if not thousands, of eligible disorders. And in my mind, this is a medical imperative. So we need to do this, and I think the question is how. And I just want to conclude with a little advertisement. Through a grant from Medicine by Design, we've formed what we're loosely calling the Together Network, which is the Ontario Gene Therapy Network, which our goal is to nucleate the community of individuals in Ontario interested in gene therapy. And we have a meeting coming up February 21st. And if anyone's interested, please reach out to me, and I'm happy to provide further details. And I just want to thank the individuals who really participated in this, our neurogenetics team, led by Ernan Gonarowski, Nana Stochitz, who's our clinical research coordinator. I mentioned Terry Pervolakis, who's going to be joining us later. Who's really an amazing force behind the SPG50 gene therapy project. And the Together initiative has been led by Kim Ambergy, who's also the project manager of Advanced Therapeutics at SickKids. And that's our neuromuscular team, and I will stop there and thank everyone for their attention.
Thank you very much, Jim. That was a comprehensive and really great tour through the work that's been done at SickKids. Really, really impressive. We are saving the questions because we're a little bit behind time for the panel discussion time. So please keep them coming in. We've got some already coming into the Q&A. In the meantime, I will hand over to Freya, who will introduce our next case study speaker. Thank you.
Thank you so much for presenting all these encouraging results and initiatives. I would now like to welcome our third case study speaker, Dr. Jeremy Duffield. Dr. Duffield is a Chief Scientific Officer at Prime Medicine. He has years of experience in drug discovery at Vertex Pharmaceuticals and at Biogen Inc. He has held leadership roles in human genetics, innate immunity, and regenerative medicine. At Vertex Pharmaceuticals, Dr. Duffield's team played important roles in discovering and advancing candidates to clinical studies in rare diseases. He had a distinguished academic career at the University of Washington and at Harvard Medical School, as well as the head of the NIH National Center for Advancing Translational Sciences at the American Heart Association-funded Laboratory for Innate Immunity and Regeneration. Dr. Duffield has received many scientific awards and holds a Bachelor of Arts, an MD, and a PhD in immunology. So Dr. Duffield's presentation is entitled Learnings from Recent Approval of Casgevy for Sickle Cell Disease for the Development of Gene Editing Medicines. With that, I will hand it over to you, Dr. Duffield.
Great, thank you very much. It's great to be here. I'm honored to have the invitation and the opportunity to speak to you today about gene editing. I've just been listening to the prior talk, a lot of information about gene therapies. Of course, gene therapies put a synthetic transgene somewhere randomly in the genome or outside of the genome. And I think those of us working in the gene editing field view the potential benefits of editing your own DNA and fixing the mutation back to the normal sequence that everyone else has as a potentially better way to be addressing longer-term genetic diseases, offering the possibility of a curative approach for those patients. So I'm going to just open up my slides. Hopefully they're going to present properly. And if they're not looking right, please let me know. That's probably on the right. Is that on full screen? Is that full screen now? Hopefully that's correct. Okay.
Yes, it's Jeremy. Yeah.
Well, let me know otherwise. Great. So thank you very much. So I was asked by the organizing committee just to really reflect on the recent successes with gene editing, and hopefully that can help to segue into what we think the future landscape of gene editing is going to look like. And I'm going to talk to you a little bit about what others have been doing, and then a little bit about what we've been doing in that to advance prime editing, which we would say is a next generation form of gene editing. So David Liu, who's the founder of our organization, who was one of the founders of Editas, CRISPR gene editing company, and also one of the founders of Beam Therapeutics that's developing base editors. He recently made a presentation and said, of course, the age of therapeutic gene editing isn't just coming, it's already here. And I think this is a theme for today's talk. And just to take a few headlines from Intellia, from Vertex partnered with CRISPR, from Verve Therapeutics, and so on. You can see that actually we now have an approved medicine, which is a gene editing medicine, with a number of programs that are in early and late stage clinical trials that have shown extremely encouraging safety and efficacy data. So just an overview here, we're going to touch on those technologies. We're going to talk about those recent clinical trials and approvals. And then we're going to talk about some of the challenges and opportunities with gene editing. And then briefly touch on one of our programs that we anticipate will shortly be starting clinical trials to treat a rare disease called chronic granulomatous disease. So just to continue on that theme, David Liu's theme, we're already here. We've just had approval of a CRISPR-Cas9 nuclease editing of hematopoietic stem cells to treat sickle cell disease. But right behind that, we have multiple clinical trials editing the liver, the eye to treat monogenic diseases, and I'll touch on one of those to treat transthyretin amyloidosis, as well as the development of CAR T cell products for oncology and autoimmune indications. And then really shortly behind that, base editors, which is a second generation form of gene editing. We have clinical trial data from CAR T cell therapy that's been incredibly positive and encouraging. And we have positive data to treat hypercholesterolemia with a once and done therapy that edits the liver to turn off the PCSK9 gene using a base editor. And then prime editing and prime medicine, which was founded only very recently. Prime medicine was founded actually in 2020. We're anticipating starting clinical trials this year. And just to put that in perspective, here's a new technology with a brand new modality for delivery that was invented 4 years ago, and we'll be hopefully testing the first patients, which is quite a remarkable contraction of the amount of time it takes to go from idea to actually having a therapeutic for patients. And I think that's going to be a theme for gene editing as we go forward. I'll explain why. So it's a little bit of a busy slide, but it just emphasizes really there's 3 ways of gene editing right now. There's lots of people in the field saying other words for these things, but basically there's 3 forms of ways of editing. One is to use a nuclease on the left, and the one that's really been advanced and is now approved is Cas9. I did emphasize there are other Cas enzymes that are in preclinical development, and there are other forms of nucleases, but essentially what happens is the nuclease finds a place in the genome, and you can see here by the schematic, it makes a double-stranded break. And then the cell repairs that break by making indels, insertions or deletions. And this is just a simplified schematic showing you the pattern of deletions and insertions you get at that place where the break occurs. So CRISPR is very good at going to that place. It's very specific for that place. But you can see it makes a mess of a gene here, frankly. So it's very good at destroying genes, but it's really not very good at all at fixing genes, particularly in the context of human disease. So very good for loss of gene function, and certain instances where you need to lose gene function occur in a rare disease, such as gain of function mutations. However, gain of function mutations are relatively rare. I think fewer than 10% of all human pathogenic mutations are gain of function mutations. Now, in the middle, what was an advance on the nuclease system was the first way to correct a gene. So on the left, very good at getting rid of a gene or destroying it, not very good at fixing a gene. Here in the middle, a base editor, which I won't go into great detail on, but is making great progress in the clinic now, is a way to fix single transition mutations. So just showing in this example, an A can be converted to a G very precisely, or a C can be converted to a T, but no other forms of mutations can be fixed this way. Now, base editors are proving to be, I think, safe and effective, but they do have the challenge that bystander edits occur. So essentially where the editor docks, if there's another C nearby, it will also correct that C or fix that C to a T, and that may be undesirable. And indeed, that can lead to loss of function as well as correction. And there's a very small editing window, which is a technical challenge. If you're trying to dock an editor here, but there's no way to dock it, then you can't get to the right place very effectively. So the inventors of these technologies worked out on a piece of paper what exactly would you need to fix human mutations very effectively in the genome when they invented prime editing. I'm not going to have time today to go over the technology in great detail, but if you're interested, our website, I'll show you in a couple of slides, has all the details of what exactly is a prime editor and how does it work. But I'm going to show you a little bit about how it works on the next slide. But I think one of the key things here is that, if we add up all of the pathogenic human mutations that have been reported, there are over 80,000 different pathogenic human mutations now being reported. They form all different types: transitions, transversions, deletions, duplications, copy and number loss, etc., shown at the bottom. And you can see that prime editors can actually address more than 90% of all of these mutations. So very different from these early technologies that have a much more limited range. So for example, these are transition mutations that theoretically a base editor could address. So prime editors can do all of these things and a whole lot more. So just to show you what prime editors can do in the genome here, these are the substitution mutations that are quite commonly found, transitions and transversions. So a prime editor can fix by creating a new flap of DNA in the genome. It can fix a single point transition to the correct sequence that you and I have without causing breaks in the DNA. And of course, it can do this in a multiplex fashion, so that which we call hotspot editing, where we can fix multiple mutations along an extended sequence. But prime editors can also fix insertional mutations by removing nucleotides, and they can fix deletion mutations by inserting a nucleotide as well. The technology has continued to advance since it was first invented in late 2019. And now we can use a dual flap technology where we actually dock two editors at the same time. And this can be used to remove expanded repeat sequences, such as those that occur in Huntington's or Friedreich's ataxia, the repeat expansion disease category of diseases. We can use this to fix much larger sequences of the DNA, which we call mutational hotspots. A single editing system will correct multiple mutations in a single region, therefore addressing many patients with a single drug. And then we can use this same technology to introduce things into the genome in a very precise and pre-specified way. And I'll show you that in the next clip here, where we can combine prime editor, which inserts a recombinase target sequence into the genome at a pre-specified place, such as a safe harbor. And then we can use a DNA donor template. Just for those of you familiar with Cre-Lox systems, this is a BXB1 system where we can actually now introduce an entire gene into the genome in a pre-specified place with a single copy. So this kind of versatility has enabled Prime Medicine to have a very broad pipeline. We actually have more than 18 programs at the company, and for a small company, that's a lot of programs. But it speaks to the diversity and the breadth of opportunity that Prime Editing can bring. And I'm just showing a few examples here. Here's a Prime Editor schematically. Oops. And you can see, for example, in our chronic granulomatous disease program, we're actually inserting 2 nucleotides into a unique place in the genome to fix the disease. Wilson's disease, a disease of copper, where we're editing the liver. We're making substitution mutations. Here, we're correcting a whole hotspot to fix a retinal degeneration. In this program, we're excising very precisely expanded repeats in the genome to correct Friedreich's ataxia mutation to the normal sequence that you and I have. And then we're using large gene insertion initially here to develop next generation CAR T products. And as I say, if you're looking for more information on the technology itself, then please visit our website. There's a movie and lots more information there. So changing topics now, I want to talk to you about the sickle cell program that's just been approved by the FDA and regulators elsewhere, including in Europe. Sickle cell is, a terrible disease, affects many individuals, characterized by occlusion of the vasculature, causing severe pain crises and infarction of organs in the body, as well as having anemia. Actually, Joe and I, Joe Mancini, who's on this call, and I were involved in this program in some of its earlier phases, and it's a delight to see this eventually coming to fruition. But just to explain briefly what's happening here, and I'll show you in the next few slides, this is a Cas9 nuclease with a targeting guide that's going into the genome of stem cells, hematopoietic stem cells in patients. And it's finding, using this spacer sequence, a gene called BCL11A. So BCL11A actually has very little to do with the making of red blood cells, but I'm going to explain how destroying this region of DNA in the BCL11A gene actually fixes sickle cell disease. So in this case, this is the BCL11A gene, and there's an enhancer region here that controls activation of this gene. And the CRISPR nuclease finds that location and destroys this enhancer so that BCL11A doesn't work anymore. And what that does is results in an increase in a form of hemoglobin that's normally turned off, which is called fetal hemoglobin. And so in red cells, one can replace the dysfunctional hemoglobin B with the fetal hemoglobin that's normally turned off. And just to show you some of the early data that were published in the New England Journal, but now there's obviously a lot more data available that's been incredibly impressive. You can see in these individuals, after they had their cell therapy product, which is an edited hematopoietic stem cell product, very rapidly the patients had a large increase in hemoglobin F, which is shown here. And the sickle hemoglobin, which is the abnormal form of hemoglobin B in these patients, decreased. So it was essentially replaced very quickly in the peripheral blood by the presence of cells having hemoglobin A and hemoglobin F. And the consequence of this is that loss of that abnormal hemoglobin B protein called hemoglobin S is that the patients who were having, prior to treatment, who were having sickling crises and coming to the hospital, very rapidly went to having no sickling crises. And so these patients have effectively had a cure for their disease. They essentially, and the data that are running out now for 2 years in many patients show that they essentially don't have sickling crises anymore. So it's been a remarkable turnaround for a disease that's really been poorly managed apart from supportive therapy for many, many years. So just to explain again how this works, because this is a real workaround. This is not a fix for hemoglobin B. The patients all have a single point mutation in hemoglobin B, but this is a real workaround, which has proven to be robust. But I think there may be opportunities in the future for a therapy that actually fixes hemoglobin B. But let me explain. So BCL11A, I explained to you there, it's a transcription factor that normally keeps hemoglobin F, the fetal form of hemoglobin, turned off. So when you once you're born and you within a few weeks of birth, hemoglobin F gets turned off and replaced with hemoglobin B. And that's because hemoglobin F has a very different oxygen absorption and release characteristics from hemoglobin B. So CRISPR-Cas9, as I explained, disrupts this enhancer and turns off BCL11A in hematopoietic stem cells. And those now edited stem cells are returned to the patient as a cell therapy product. And when those hematopoietic stem cells now make erythrocytes, those erythrocytes have a permanently activated hemoglobin F, which now compensates for and replaces that dysfunctional hemoglobin B, which is also called hemoglobin S. And so you can see here what the way it works is the patients have harvested CD34 stem cells taken from the bone marrow. Those are edited outside of the body. The Cas9 editor disrupts that enhancer, turning off BCL11A, and those cells are then returned to the patient as a cell therapy product that they enter the bone marrow and take up residence in the bone marrow. This approach does require conditioning of the bone marrow to create the space for those cells to return and to populate. So changing gear now, just to talk about liver programs. And there's been a lot of advances in delivering gene editing technologies to the liver. We mentioned Cas9, and Intellia has really led the way in developing an RNA lipid nanoparticle system to deliver to the liver, very much like the vaccines from Pfizer, BioNTech, and also from Moderna that were a lipid nanoparticle encapsulating an RNA. So building on that technology, I think we're going to see a lot more of this in the future. You can see Intellia has developed an mRNA form of the Cas9 nuclease and a guide RNA that goes inside the nanoparticle together with the mRNA. This is delivered by an intravenous infusion, and it goes to the liver. It's taken up by hepatocytes in the liver, and you can see now that mRNA is made into a protein, the SP-Cas9 enzyme, by the liver cells themselves. It now finds that guide RNA that was co-delivered, enters the nucleus, and there it edits. And in this case, Intellia has been disrupting the TTR gene to get rid of transthyretin in the bloodstream to treat patients who have a disease called transthyretin amyloidosis. So it's really a remarkable story of using RNA encapsulated in lipid nanoparticles to deliver next generation gene editing drugs to the liver. You just heard from the prior speaker about using viral systems to deliver to the liver, and they certainly have had some toxicities associated with them. And I think there's a lot of anticipation that an RNA LNP way of delivering these next generation drugs could prove to be safer. So just to show you some of the data from Intellia, the goal here was to disrupt the production of transthyretin, which is a highly produced protein which is in the circulation and can deposit in the heart and peripheral nerves to cause disease. And you can see in a primate study, a preclinical primate study, they were able to show in a dose-responsive way that when they delivered their drug at 1.5 mg/kg or 3 or even 6, they were able to get a dose-responsive reduction in the concentration of the transthyretin protein in the circulation, which is again released from the liver and is a normal part of the circulating proteins in the blood. And you can see a very, very high level of reduction of transthyretin in the bloodstream from these doses. And one of the delightful things, I think, for the company and also for the whole field was that when the first patients were dosed at 0.1 mg/kg and then at 0.3 mg/kg, which is significantly lower than in these NHP studies, they were able to see a robust reduction. Here's the first 3 patients treated with this, and you can see within 14 days, they had a very robust reduction in transthyretin in the bloodstream. And this program's continued to prove to be safe and efficacious, and is at least in the phase 2 studies, and is now in phase 3 clinical trials. So I would just add that there are additional trials from Intellia that some of the base editing companies have started clinical trials and even already shown early clinical efficacy for base editors for treating hypercholesterolemia. And Prime Medicine has developed a prime editor delivered with a nanoparticle system that looks very similar to this to correct genes in the liver to treat diseases. So we anticipate that there'll be a good number of trials where we're treating diseases by targeting the liver. And indeed, we think there are hundreds to thousands of diseases that could be targeted by treating the liver. So just to broaden that discussion a little bit further, I think one of the challenges for gene editing technologies is the delivery. Of course, the gene editor is an RNA and an enzyme complex together, a ribonuclear protein complex. These are very large molecules. They have a lot of charge on them. You can't swallow them and they get absorbed through the gut and end up in your cells. You actually have to work out clever tricks to get them into cells, to get them to the right parts of the body. And of course, even when they get into the cell, they've got to get into the nucleus in the right concentration to do their job. So very different from small molecules or antibodies. The whole paradigm of drug discovery is kind of turned on its head. I think one of the other things about editing technologies is that they're once and done. And so that brings a lot of opportunities. If you're able to deliver them at the right concentration and fix the genome back to the normal genome that you and I have, then it's essentially a genetic cure for those patients, particularly if you can address them early enough in their disease course. So Prime Medicine has developed 3 different approaches to deliver the drug to patients. One of them, like the sickle cell program from CRISPR Therapeutics and Vertex, is to edit cells outside of the body and return those cells to the patient. We're using that for our own hematopoietic stem cell programs, and I'm going to talk to you about one of those in a moment. We're also developing CAR T cells, next generation CAR T cells with this approach. And I think this is proving a very robust approach, although it has some challenges of obviously the expense and time of extracting cells from patients and putting them back is not inconsequential. So we and others are developing approaches to try and minimize the hurdles that are required for these patients. I just highlighted Intellia's progress and the base editing program's progress with LNP RNA therapies. So we've developed prime editors for liver programs and lung programs. And I think we're anticipating extending the LNP technology to deliver to other places in the body, including leukocytes and potentially other organs, including muscle and CNS. And then, for right now, we're also developing AAV therapies where we can deliver the prime editor as an AAV system. One of our most advanced programs using AAVs to deliver is to correct mutations that cause retinal degeneration by delivering the AAVs to the eye. But we're also using these to develop lung and neuromuscular programs. And so, today I'm just gonna, for the sake of time, focus on the ex vivo cell therapies. This slide just talks about, I think, the opportunity with gene editing. Yeah, if you think about how do we make a small molecule drug, we find a target. We hope the target's well validated. There's whole teams that work on validating that target. Is it a good target for the disease? They're not usually the cause of the problem. They're usually some downstream component of the disease, and so the efficacy of that drug, even when it works very well, is often limited. And of course, then every time you want to make a new drug, you got to go back to the starting line again. And I think for those of us who worked in small molecule drug discovery, it's it's finding a needle in a haystack. The chance of success is low, and toxicity, unexpected toxicity, is often a problem later on in the drug discovery process. So the advantage of gene editing is we often know the cause of the disease. It's often monogenic. We can be absolutely sure that if we can fix the problem, that we can have an impact on the patients. So that's one huge benefit. But I think the other benefit we're learning is that this is a very modular system. It's a little bit like an app on your iPhone. If you've got yourself an iPhone, that's your delivery system, then you just change out a guide sequence, which is a Watson-Crick base sequence, and you can have a whole new product. And so this leads to a very modular way of building drugs, where we essentially take components that are already fixed and the FDA's seen and they're approved, or other regulators have seen and approved, and we can essentially swap things in and out and potentially have a new drug very quickly. And indeed, that's proving to be the case at Prime Medicine, that I mentioned that within just a few short years, we've been able to build one of the first drugs, and we're anticipating many rapidly following candidates to come right behind that. So in the last few minutes, I'm just going to talk about one of our programs that we're anticipating to start clinical trials this year. This is a disease called chronic granulomatous disease. This is a terrible disease. It's one of the first genetically defined diseases. I think back in the early 80s, the gene that caused this disease was first described, and the gene, actually the cluster of genes that encode this enzyme complex, NADPH oxidase. These patients get recurrent severe infections that are often lethal. And even with modern medicine and modern antibiotics and intravenous infusions and intensive care units, the patients often do not live well into adulthood. And even today, sometimes the disease is lethal from infections early on. So it's a really awful problem. And the reason they have this recurrent infection problem is that the neutrophils in the circulation lack this enzyme complex that the neutrophil phagosome uses to produce oxygen radicals to kill pathogens. So the lack of this enzyme complex means that pathogens can evade the innate immune response. So many of the patients have a single mutation in this protein here, the P47 protein, and it's a loss of a G and a T in one of the exons. It puts the gene out of frame, so there's no protein produced. So prime editing is the only technology we think that can fix this problem, where we can insert very precisely that G and T back into the gene to recode the gene so that it's the normal gene that you and I have. Our approach is to take the patient's own stem cells out of the body, much like the Vertex CRISPR Therapeutics program has done for sickle cell disease, insert that G and T back into the NCF1 gene that encodes this protein, and fix the protein so the protein works completely normally in the patient neutrophils. And so this is just to show the genotype here. This NCF1 gene in you and I is completely normal. The gene locus is very complex. It's actually flanked by two very funky pseudogenes that are identical to the NCF1 gene, but actually also have this GT mutation in them. And that turns out to be a real advantage for us, because we can actually fix one of these, or we can fix this in the patient, which is shown here as the patient. The patient has these abnormal loss of G and T here. So our prime editors are putting the G and T back into these genes, but they can also put it back into one of these genes to fix the disease as well. And so very similar to what I showed you for Casgevy, we're harvesting the patient's stem cells from the bone marrow, CD34 cells. We're editing them outside of the body with a prime editor that corrects that G and T by putting precisely that G and T back in. So this is the normal gene that you and I have. We cryopreserve those cells and then return them to the patient as a cell product. And just to, I think this is very quickly, I won't go over this, but just to show you this is what the patient has. And we've demonstrated very clearly that we can fix one copy of the NCF1 gene, or we can fix one of the pseudogenes, and that is necessary. That is sufficient to correct the function of the NADPH oxidase completely back to normal. And just to show you a little bit of preclinical data from this program, these are patient cells, patient CD34 cells. We deliver the editor as an all RNA product, again, a little bit like the COVID vaccine, but here we're not using a lipid nanoparticle. We're delivering this as free RNA. And you can see that the patient cells are very efficiently corrected with a prime editor. Here, 80% of them are corrected. The remainder are untouched. We don't have any mistakes when we make this edit. And then if we differentiate those into neutrophils outside of the body, you can see that 80% of them now make that protein. And then when we test the function of that enzyme complex, they now make a normal level of oxygen radicals from the NADPH oxidase enzyme. So, you know, really remarkable data that, you know, connect the genotype here, the amount of correction to the amount of phenotypic function that's restored in the patient neutrophils. You know, one of the challenges of editing stem cells outside of the body and putting them back into the body is it's a complex process. We know that CRISPR and Vertex have been through this recently, but when you harvest CD34 cells, they're not actually one cell, they're actually multiple cells. There's the true long-term hematopoietic stem cells, but there's short-term stem cells and there are committed progenitors in that pool. And so when you edit outside of the body, you've got to make sure that you edit these and you don't damage them in the process. And of course, these are the few cells that permanently make all of your blood for the rest of your life. So you want to make sure you've treated them very, very gently and that they're going to completely function normally. So there's a lot of rigorous testing that has to occur preclinically in order for us to be sure that our product is completely functional when we return it to the patient. And one of the experiments that we use is we engraft edited cells, which is all of these, but we put these into an immunodeficient mouse model, and we wait for 4 months, which is this time point here, so that those long-term stem cells can repopulate the bone marrow and make all of the blood cells, and all of the short-term cells are used up. So this is a very key time point for us to evaluate the function of our edited cell product. So just to show you a little bit of data from those experiments, and, you know, we think this is really differentiated. You know, prime editors don't break the DNA. They don't damage the DNA as far as we can tell, and I'll show you a little bit more data in a moment. And so what we see is that actually we get almost 100% engraftment of the bone marrow. And importantly, it's no different from unedited human stem cells. So if we just take unedited cells and engraft the mouse bone marrow here, with this process, we see almost 100% engraftment, and it's identical following editing. And then when we look at those long-term cells after 4 months, they continue to hold the corrected edit. So there's no loss of the edited long-term stem cells in any of our experiments. So we're highly encouraged by these, and this is just you know, a snapshot of a huge trove of data we've generated for this program. But, you know, just to summarize, you know, we can highly efficiently edit the patient stem cells, that the long-term fraction are highly efficiently edited, that they have all the normal functions that we would expect, and they behave normally and make all of the blood cells in the bone marrow. So these kind of studies are very encouraging and, you know, enable us to hopefully advance to clinical trials very shortly. I'm just going to mention one really, really critical area, and it's certainly an area that regulators have paid a tremendous amount of attention to, and that is that when you're editing the genome and it's permanent, what about all the other things that might be happening in the genome that you don't know about? And so there's been a tremendous amount of work done to really understand what was called off-target editing. So are you editing elsewhere? Are you causing breaks in the DNA and causing translocations or deletions? And certainly, I think the more we've looked at this with the CRISPR nuclease system, where a break in the DNA happens, if you imagine there are other breaks happening in the DNA, you can get all kinds of strange things happening in the DNA. And there are certainly some concerns about long-term safety, although I think certainly for the sickle cell program from Vertex and CRISPR, I think right now things are looking very, very good. But nevertheless, this is a really important area to think about safety and patient health. If we're inadvertently going to cause patients harm down the road, then we certainly want to know about it. Now, we think prime editing is differentiated. It does not break the DNA. It creates a single-stranded nick, which is shown here. Here's a prime editor engaging with the DNA that has a mutation in it. The prime editor creates a nick in the DNA, opens up a flap, but it never snaps it. And then the other additional thing is, in addition to only creating a nick, there's 3 places where the prime editor needs to interact with the genomic DNA in order to make an edit. So here's one. The next part here is that actually we have a sequence here that binds to the 3' flap, and then the 3' flap, which is recoded now, interacts with the genome as well. So that's 3 steps where you have to get Watson-Crick base pair annealing to occur in order for editing to happen. So because of these 3 steps, as opposed to 1 step with a nuclease, we think that prime editors are much less likely to cause off-target edits. And indeed, the data we have so far is incredibly encouraging. So just to talk briefly about this, we've developed a suite of assays. I think they're the most comprehensive suite of assays that are actually being developed yet, but they involve a number of assays to identify at-risk sites in the genome, which are shown here. And then we do very deep sequencing of those sites to identify off-target sites. And then we have a suite of assays looking at the risk of translocations or large deletions or other funky integrations that could happen in the genome. And I'm going to show you some data on those in the next slide. So this is from our chronic granulomatous disease program, and here are a snapshot of some of the data. And the headline is that actually we haven't found a single off-target site in the entire genome following prime editing for this program. What we're showing on the bottom left is actually for our 4 lead programs. We haven't found a single off-target site for any of those programs at this time. I think that's really very, very different from the data that you can see in the public domain from CRISPR-Cas9 systems. So just to show you this, this is 550 off-target sites spread across the x-axis here in the order of the chromosomes. These are at-risk sites that we identified with our assays. We then deep sequence with more than 1,000-fold deep sequencing. And you can see we don't have any off-target editing identified at any of those sites, but we can regularly find reliably find the on-target edit site shown here. This is a set of assays now, a one-ended PCR-based assay to look for translocations and deletions that could occur when the prime editor engages and nicks the DNA. And you can see that in patient hematopoietic stem cells that actually we do not find any evidence of deletions anywhere in the genome, and similarly for translocations. And just to show you a positive control here, if we do exactly the same experiment with the same guide RNA sequence that CRISPR-Cas9 uses, and we use Cas9, we can find over 10% of hematopoietic stem cells now have translocations in their genome. So, I think these data are, incredibly encouraging. Obviously, the proofs are going to be the continued evaluation, but certainly for our lead program that is now, approaching clinical trials, we haven't found any off-target editing. Just last but not least, I'm here representing the company. Prime Medicine's based in Cambridge, Massachusetts. We have about 250 people at the company. I would just say I'm blown away by the people I'm working with. They are some of the smartest, creative, diligent, energetic people I've ever worked with. It's just a delight to be working with this team. And just to show you a little bit at the bottom here, we're not, we are a fully industrialized company. We're making all of the drug components ourselves internally, and then we're partnering for GMP production, but we're going all the way to GLP production ourselves. And I'm happy to talk more about that, but we think that's a really important core for us to really understand our drug components extremely well all the way to the clinic. So I'll stop there, and I think there's no time for questions, but happy to address questions later on. Thank you.
Thank you so much for this fantastic talk. There are some very interesting questions from the audience already, and we invite the audience to keep adding questions for the speakers and the panelists in the Q&A box at the bottom of your screen. Your questions will be answered during the panel discussion, and that will follow the keynote session. So we would now like to take a brief break, and we would like to be back in 5 minutes for the keynote session, which will be presented by Dr. Frappier. So we will see each other around 3:12 Eastern Standard Time. Thank you.
Hello, everyone. Welcome back after the break. We're going to make a start on the final speaker of this main session of the symposium. So I would like to welcome you, Fiona, Dr. Fiona Frappier. Dr. Frappier is responsible for developing regulations and guidance for industry, all while improving stakeholder outreach. She is working on new regulations that will support a modernized approach for regulating clinical trials and is Health Canada's chair for the rare disease cluster. Dr. Frappier also manages stakeholder communication on complex product approvals and ensures timely decisions and coordinated communications. Dr. Frappier has 14 years of experience at both Health Canada and the Public Health Agency of Canada, working on interdepartmental science policy issues. Prior to this, Dr. Frappier completed her PhD in microbiology and immunology at the University of Ottawa. Dr. Frappier's presentation is entitled Regulatory Framework for Rare Disease Therapy Development. And with that, I'll hand it over to you, Fiona. Thank you.
Thank you. And thank you so much for organizing this symposium today. It's been really a very impressive afternoon already. And so it's wonderful to come and speak about regulations and how this fits within the larger Canadian and global context for getting these to the patient now. Oh, here we go. So I think when we get questions at Health Canada, we often get asked a lot about how Canadian specific are your rules. And I would say generally, we endeavor to never have Canadian specific rules. Drugs are developed under a global landscape. We make every effort to coordinate with Europe and the United States and the larger global context to ensure that the rules are harmonized and reasonable and are being applied in a fair way. This is very important because we represent a small percentage of the global marketplace and import the majority of our drugs, which are actually manufactured outside of Canada. Once you get a Health Canada authorization, I was asked to speak to the interplay between federal and provincial governance. I'll highlight that that's not where the story ends. What that means is you now have an authorization to sell your product on the Canadian market. There's a few more steps towards when it actually enters into a hospital. The first one's very important. It's called a health technology assessment. This is where we look at the overall value of the product as compared to what's already on the market. There are two organizations in Canada who do that, in CADIF and in Quebec, INES. And really what they're assessing is it a good price point for the efficacy of the drug, and would we recommend a reimbursement? We also have another regulatory body that looks at price protection. And the idea is that given our very generous patent system, we want to make sure that we're not creating monopolies where the pricing is unreasonable. And so if we think there is a case where that may be, they get referred to the Patent and Medicines Review Board, and they take the case on and they look at what is the data and what does it mean. For gene therapies, there's a fourth level to think about, and this is the price negotiator. And so there's a few products, they don't do all of them, they do maybe a dozen a year, where the Pan-Canadian Pharmaceutical Alliance gets together. And normally, actually, each province, all 13 jurisdictions, negotiate their own prices in board, and they actually operate separately on a provincial scale. There's a few where they come with a national approach, and they're usually these very high-cost products to make sure that there's a fairness. And it's also to help support avoiding a future postal code lottery where it's reimbursed in one jurisdiction and not in the other. This then leads to another layer, which is, do you get listed on, in Ontario, where I am, on your OHIP plans, your provincial reimbursement, or do you get reimbursed on your private plan? And so when we're thinking of some of these data sets, it's important to look at all the people who will be asking for data from your product along the way, and to be making sure that we're developing a research program that will eventually feed into a larger systemic piece. Again, we do this in conjunction with our international colleagues. They have very similar systems set up, although operate differently, of course. I think one of the things we get asked a lot in Canada is, do we even have rare disease therapies? How many are these on the market? I would say that this has been for multiple years now, almost half, if not over half, the products that we're approving every year for our new active substances. So this is, very well represented. It's a large scale endeavor. Of those, a few of them are gene therapies. This is new, the CAR T cell therapies, as well as the viral vector therapies, which we've heard a lot about today. What does that mean for the regulator? And so I think there's a few key interactions with us. So one, we've heard about, especially today, which is the pre-market. And so as we're developing our animal studies and our first in human trials, what does that look like in terms of the data that we need? Then we enter into initial clinical trials, which does get filed with Health Canada. This is under Division 5 of our regulations. It's actually not that terrible to fill out in terms of forms, but you do need to have quite a good, strong data set to justify the trial itself. Once you get the form to us, it's also not that big of a deal. It takes us 30 days to review it. We guarantee a decision within 30 days, and there's no fee of any kind associated with it. This is unique in the global context. Most other regulators do charge. And I think for this audience, what I would also like to highlight is we have an additional form called an OLIP. It's an open label individual patient study trial. In some rare diseases, this is actually very appropriate. And so, if you're ever in that situation, to ask us about it. All these forms are online. I'm going to share them with you as we go. The next phase is when we get into really what is a pre-market submission. This is when you're looking to authorize the sale of the drug. There's quite a number of different regulatory requirements. Notably, the registration of the sponsor themselves as a company is liable for the information that they're sharing with the public and the prescriber. There's the establishment license itself. And so this is making sure that you have a license for a commercial grade product and a good manufacturing practice. So we can say that you're consistently manufacturing the same quality every time. For biologics, we also look at Division 4, which is regulating a lot of the biological components. And so what you'll see there is really an acknowledgement of trend attributions in the manufacturing processes, which are different than pharmaceutical or organic compounds. And all of this culminates in what we call Division 8, which is the new drug submission pathway. And so you would come with quite a large package of information, many trials, all of the patient information, and we would be evaluating 3 attributes: quality of the manufacturing itself, and looking at the ability to manufacture consistently, batch over batch. We'd be looking at the safety of the product itself. And so this includes, as we've heard today, looking at, other organs and how they're impacted. It can be looking at mild side effects, just fever or a rash. It can be looking at severe outcomes such as death. And so it's categorizing those and making sure that we're accurately narrating the impacts to the patient. And this will eventually inform the prescribing information on the product itself, and then the patient acceptability. And the third thing we look at is really the efficacy of the data itself. This can be biomarkers, this can be long-term outcomes, it can be short-term outcomes, it can be dependent on different designs. The trial size can vary. We see a lot of small trials with rare diseases. This design component can be proportional to the product that's being proposed. But at the end, what we do is we say the totality of the evidence across these 3 parameters. We can say the benefits outweigh the risks for the product. And then we issue what's called an NOC, a Notice of Compliance. And this gives you the right to sell on the Canadian market. Once you're on the Canadian market, we actually continue to oversee the product. And so this is the post-market oversight of the product. For biologics, it can include lot release. So a lot of the cellular therapies, the lot itself is released and evaluated. It also includes adverse reaction monitoring. Even though it was characterized in the clinical trial, a clinical trial will really only see a small percentage of adverse events. Once it gets given to a larger population, you're going to see some exceptionally rare adverse events. We need to continue to record them and update the post-market product monograph. And then if there is a case where, we heard an example today where we can see there's a liver damage, we'll implement a risk management plan. And that can include something as simple as liver screening, where you say the person, in order to be eligible for this drug, actually has to have no pre-existing liver function issues. Again, the benefits and the risks will need to be evaluated, but this is what a risk management plan is looking at more complex situations. And then we have inspections, and this is our sister branch. So this is the operations section where they actually go to the manufacturing sites themselves. You know, it could be 5-6 years after approval and just, you know, do a general audit of the good manufacturing practices and the records to ensure that everything is in order and that there's no issues. I know it sounds like a lot. So we have online a roadmap. It's a regulatory roadmap for biologics. We also have one for rare diseases, and it has every little form, every little associated guidance document. I will warn you, there's quite a number of them. I'll also say for scientific communities especially, they're also generally principle-based. And so you won't find within these documents, you know, it has to be 10 milligrams of this in order to be compliant. They're not specified in that way. What they speak to is the principle that we'll be upholding. And the idea behind that is to encourage innovation. So there could be different ways of approaching a problem to arrive at the principle that needs to be demonstrated, and that's okay. And so you'll read through these guidance documents and get that flavor. And they go across, you know, the key elements, which is really that clinical trial phase, which can occur for multiple years, the new drug submission, looking at the good manufacturing practice and getting that establishment license, as well as the post-market requirements and then post-market updates that will occur. The other question we get asked a lot is, do we fast track drugs, and what would that look like? And do we encourage drug development, and how do we do that? And so there's a few key things to know about. The first one is we do have pre-submission meetings. This can be prior to filing a clinical trial or prior to filing a new drug submission. And in these meetings, we do encourage, you know, the sponsor to write out questions they may have or even options they're considering, and we'll provide, you know, advice on how compliant we think those options are, or if, you know, they would not be acceptable as proposed. And so that's good information to get before you even file. And then, you know, some of those key juncture decisions. We do on occasion have multiple pre-CTA meetings with a sponsor if it's needed, in order to make sure that everything will go well once they come in. Once the new drug submissions come in, we do have priority reviews. This is a reduction of quite a number of days. A normal review is 300 days. These are huge packages. A priority review will come in 180 days. And this is really because they're offering a substantive improvement over anything else on the market. And the idea is that it will reduce the time to patients who really need it. There's another pathway that's important to know about. It's called a notice of compliance with conditions. And this seems longer because it's 200 days, but they can actually come in with less substantive evidence, where we still have questions about the efficacy, and they get a right to sell on the market, but have to make a commitment for a confirmatory trial. And so every product that's approved like this, it's noted in their product monograph. It's also noted on our website what they've agreed to in terms of confirmatory trials and ongoing work. But in these cases, it's not only promising evidence, it's also, you know, dealing with really nothing else on the market and quite severe outcomes expected without it. And so this is where that balance of benefit and risk is considered to favor towards waiting for the confirmatory trial once it's on the market. In addition to this, as I said at the beginning, our HTA, our health technology assessors, Kadith and Ines, also do a review. And so instead of waiting for a decision from us to then begin a review with the HTA, you can apply to have aligned reviews. And so the packages are going through at the same time. This can be very influential in getting the product to market quickly, assuming that both packages are ready for review. And the other thing we do is a use of foreign reviews. This isn't quite the same thing as a use of foreign decisions, but it's where we say, you know, it's quite a complex package. We'd love to see what the EMA or the FDA did and how they deliberated on it. And we can refer to their reviews in informing our decisions and looking at how we think about it. And this can actually save a lot of time for our scientists, and we can connect and see some quick follow-up questions to keep products moving. This assumes that they've already reviewed it. Sometimes we actually receive the submissions at the same time, in which case it's more a live interaction than referring to documents. I think the other question we get asked quite a lot is, what does this mean for the number of new active substances on the market? This is old data, but it was done by a really nice group called the Center of Innovation and Regulatory Sciences, CIRS, and they really looked at year over year between the EMA in Europe, the FDA in the US, PMDA in Japan, ourselves, Swissmedic in Switzerland, of course. What are we looking at in terms of the overall numbers? And then what is the percent of orphan drugs? And I think you can see that in Canada, we're approving the same number of new active substances, and it's reflective of the percent submissions that we see. And I know we do get asked a lot about what does that mean for new drug submissions. And I think we do see once the new active substance is on the market, there isn't as much what we call supplemental drug submissions. And this is for every other indication that gets led onto it. And so there you see quite a significant number change and some off-label use in Canada. And so there's some things to think about there in terms of future regulatory actions. And then today we also talked a lot about the viral vectors. And so I just thought I'd pull up here for us. Some of you already know all of this by heart, but looking at the numbers, what has been improved in Canada, what vector systems are they using? You can see here the AAVs, the lentiviral, the retroviral. What were the dates? Some people are very interested in looking at the dates in Canada versus the other larger markets. And then as we were talking about the accelerated pathways, how many of them got NOCCs? 3. And how many of them got priority reviews? 4, but not all of them, right? And so that speaks to the clinical packages as well. And then in terms of volume of clinical trials, to give perspective in Canada, we would authorize, well, we would issue no objection letters for a new trial to start. About 3,000 new trials in Canada every year. Since 1994, 300 of them are gene therapies. So I think it gives you an idea of the scale of this work. And then I do want to speak a little bit to how we organize our work with international counterparts. And so there's been significant work done on how do we want to look at assessing cell and gene therapies, how do we want to look at managing GMP, how do we want to look at managing efficacy in rare disease trials. And so some of the key organizations to know about, all the guidance documents published online, is the ICMRA, ICH, and PICS. And so these are the main consensus bodies where we publish policy statements as well as guidance documents that all the international regulators can agree to. And so they're quite influential. And then in terms of work sharing and looking at how do we advance parallel reviews, I think some of the key organizations that we do are rare disease cluster meetings. And so this is an almost monthly meeting that Health Canada has with the EMA and the FDA to look at the pipeline of what's coming in and what should the approach be, what are some of the things we're worried about, and have a little bit of a pre-conversation about what that looks like. The other one is the EMA Open, and this is an opportunity. It was done quite extensively under COVID. It's a new opportunity for rare diseases where we can look at co-reviewing and sharing that review strategy. We also do that with what's called the Access Consortium, which is Australia, Canada, Singapore, Switzerland, and the UK. The Access Consortium is an important one because it's not just looking at what do we do for shared review, it's also trying to create a bit of a pull market where the 5 of us together create quite a larger market. And so it incentivizes an earlier application to all 5 of us in a coordinated way. And then lastly, it's primarily oncology, but actually that's quite a lot of rare disease drugs, is Project Orbis. And this is where we can look at really much earlier reviews and coordinated information sharing. And so these have all been very effective and are continuing as we go. I think in terms of resources, we should think about it in a few key ways. So one is just the guidance documents online, familiarizing ourselves with them, looking at different components depending on the product line itself. I think there's also many multiple communications. So there's communications with external stakeholders, with our sponsors, with our other regulators, and making sure that there's actually quite a lot of information sharing. With gene therapies especially, but cell and gene therapies, there's often also classification issues of which rule will apply in this circumstance. And so that is, that's an international endeavor to keep sure that we're classifying in a homogeneous way that is fair and that will advance the industry. And then, like we said, it's about ensuring that there's good harmonization and not overly specific Canadian rules. And if there is, it's because it's actually very justified, well explained, and there's a publicly facing guidance on why it will be approached in that way here. And that's it. It's open for questions, although I think we're actually moving into the panel right now.
Thank you, Fiona. That was really clear. Thank you very much. I will now just move on to the panel section and introduce Dr. Doreen Wong-Reger, President and CEO of the Canadian Organization for Rare Disorders, to moderate the panel discussion. Dr. Wong-Reger is also the chair of the Consumer Advocate Network, President and CEO of the Institute for Optimizing Health Outcomes, and the chair of Canadian Health Patient Alliance. Internationally, Doreen is chair of the Rare Disease International, president of Asia Pacific Rare Disease International, treasurer of the United Nations Non-Governmental Organization for Rare Diseases. She's also chair of the Patient Advocates Constituency Committee of the International Rare Disease Consortium, patient advisor to the APEC Rare Disease Network, and a member of the editorial board to the Patient-Centered Outcomes Research. Doreen has a lot of different achievements, extremely impressive. She's also served on numerous health policy advisory committees and panels, and is a member of the Genome Canada Steering Committee for the Rare Disease Precision Health Initiative. Doreen has a PhD in psychology from McGill University and was a professor at the University of Windsor in Canada. That, I'll hand it over to you, Doreen, and thank you very much. And I'll ask also our other speakers to join us. Oh, you're on mute, Doreen.
If we could get all the other speakers that are on this panel to turn on their camera, that'd be great. Thank you very much, John. I don't know how we're actually going to do this, because that was such an amazing tour de force in terms of presentations. It's certainly for us that are not deep scientists, this was really more than a masterclass in terms of gene therapy, and certainly not just the science behind it, but also the applications to it. What we want to do in this panel is hopefully to be able to address the different kind of questions that have come forth, but also to move the discussion, I think, into where some of these questions are, and that is, what about implementation in Canada? So I think what Fiona just presented was a huge, I think, help in understanding what is the processes that we go through, and certainly some of the ways in which Health Canada and the other agencies are involved. So we have some technical questions here. I may try to mix back and forth between some of the technical questions and some of the other more applied questions. But first of all, I would like to introduce, you met the other speakers, Jim Dowling, Jeremy, who just had spoken just before this, Jeremy Duffield, and Maria Moreno, who spoke earlier. But I want to introduce Terry Privalowski, who actually Dr. Dowling referenced, and really referenced very strongly. He is the father of a child with SPG 50, and he and his wife really went through a great deal of initiative on their part to actually raise the funds and to commission I guess a N of 1 gene therapy trial, and successfully in 3 years' time, with the support of Dowling and his team at Sick Kids, came up with a gene therapy for his child and for a couple of other children that also had the same condition that I guess they discovered along the way. So it's an amazing story about how the collaboration between certainly a very committed family and the very willing scientific community and very supportive regulator in the background there to help us to get to this amazing achievement. And out of it, I think we've also then had Terry, and again with some of the collaboration, I think with regard to his co-investigators, I guess, have been able to put together a consortium or called Alpida, which is not only going to be using some of the work that they've done in terms of developing this gene therapy, but also helping other parents, families that are interested in going through that same route, which is amazingly valuable. So just wanting to kind of introduce, I think everybody else is still on from before. Maybe I'll just start with a couple of somewhat technical questions, but I'm going to ask people to kind of really keep their responses somewhat high level and brief, and we will give you an opportunity later on if people want to contact you specifically for much more detailed discussion to do so. So one of the questions that came up, I think one of the early ones that went over to Jim, I thought was worthwhile. It says, you talked about the fact that in the research that you're doing with the gene therapy, discovered and previously unappreciated, I think it's what it's called, liver genotype for XOMT-MGT that caused multiple serious adverse events. That was very challenging in terms of continuing the trials there. Is this something that suggests that one needs to look at, if we're talking about a gene mutation, all the possible manifestations it might have in terms of proceeding with a gene therapy, or was this something somewhat unique in this situation? And if one needs to look at all these, how do you go about doing this?
Yeah, I guess I can start. It's a great question. And I think it's uncertain whether this was a unique occurrence with this particular disorder or whether it's something we need to be attuned to for other conditions. And we see many diseases that are caused by mutations in ubiquitous genes that have very specific organ manifestations. And there's always possibilities that other organs may be involved, and that can come out with exposure to experimental agents for sure. I think some key challenges are the fact that animal models are not humans, and we often rely on what we find in preclinical models to inform on not just effectiveness, but also from side effect profile. And I think the liver disease is a good example because the preclinical models didn't originally show any liver disease. So that's something that we can't rely exclusively to say, well, you can't have problems if they weren't seen in the animals. And then I think maybe more broadly, the issue would be to just have that in the back of your mind when doing the lead-in studies to help inform on interventional studies related to the natural history studies and opportunities there. And I guess maybe it takes some not ignoring of signals that might be percolating in the background. And I think, if we look in retrospect with MTM, there was some evidence that was presented to us that there might be liver involvement. But I think, given all that we knew, it didn't seem like something that required perhaps real precise attention. And so maybe it's just being very alert to what biology is telling us and what patients are telling us. Particularly in informed natural history studies.
Yeah, and I think that's the challenge sometimes, so in terms of being able to have good natural history studies when we're talking about some of these rare or ultra rare conditions. So, I mean, Terry, I think when you went into this, right, was there any indication from you folks as to what that natural history might be and things that, working with Jim's team that you might need to be alert to?
We were very fortunate that our disease was very studied. The base biology was studied for almost 20 years. There was a natural history study that was being run at Boston Children's. So we were very fortunate in that perspective. But I think, for example, one of the other programs that we took on is called CMT4J, and it doesn't have a natural history study. And it's almost like 4 diseases in one. It's a very diverse disease, and it makes it extremely complicated in order for us to study and create a therapy for, because of diversity of within the disease itself.
And that's a big challenge. I think we all know is that diversity in terms of the diseases, even though it may be either a single gene or a small cluster of genes that actually cause it, it can actually show up quite differently. And maybe, this goes back, now just make the comment, Chris McMaster alluded to it earlier, and that is, how do we set up in Canada more of these patient registries so that we can actually start to collect these natural patient, histories, because without that, when we come to the opportunity of maybe being able to introduce a therapy, we don't know enough. And I think, Fiona, maybe you can comment, to what degree is Health Canada quite willing to look at, natural history studies as being an appropriate, a replacement for maybe a placebo-controlled trial? How is Health Canada using that?
So you're right. We do externally controlled as well as internally controlled natural histories. It's a complicated question. Normally, we would assume that you would conduct the natural history before designing the mode of action to approach your clinical intervention. So I think that can be a bit complicated when we start looking at, as we heard today, sometimes there's a very divergent presentation. There might actually be multiple genes involved. And so when you start evaluating the clinical trial, it gets very messy if you don't have a clear baseline. And so you can actually, in some cases, waste a couple of years that would have been faster had you completed the natural history first. And so some of that becomes a bit of a business decision, but I can say we do review drugs that have ongoing natural histories that are concurrent to the clinical evaluation. It just doesn't always save as much time as you would think.
I could make one comment on that, because this is we're going to see more and more of this challenge as we try to advance precision medicines for small groups of patients. So that there are two approaches. One is a prospective natural history study where you say, this is what we're going to collect and we're going to go and do it now. And then potentially those patients can become eligible for a trial. And then there's the retrospective where you go into hospital databases and collect them patient data. I think the challenge with the latter is the consistency of the data. And so one potential way to get around that is to actually provide guidance to physicians. If we don't have the resources to run a natural history study, perhaps if we're standardizing the kind of information we're collecting along the way, just as we see these patients, that might help with robustness of the natural history data that we can collect quickly when we decide to move forward with a new therapeutic.
Yeah, those are really great guides, and certainly as we're implementing some of the research that's coming in, especially with the rare disease drug strategy and some of the programs that Chris is talking about, those would be tremendously valuable. But again, this is something going forward, and again, that will still take some time. But I think appreciate the fact that in Canada we're really trying to get into the game. I think we always kind of felt as Canadian patients that this was working around us as opposed to with us. I'm going to switch gears just slightly, and maybe this is something that both Jim and Jeremy, you can talk about. There was a question raised in here that asked the question of, what do you see? I mean, it was I think to you, Jeremy, but maybe Jim can answer as well. What do you see as the major advance to move away from AAV and to in terms of vectors and using something more like the kind of gene editing approach that you're using now, or next generation delivery technologies?
Yeah, I could maybe take a stab at that. I think what we've seen, and Jim presented some incredible data that's powerfully helping patients. So those gene therapies where we put in a construct that humans have made that's similar to the gene, that's with a synthetic promoter and often lies outside of the genome. In many cases, it's helping patients. There are other cases where it's not helped patients nearly as much as we would have expected, or hasn't helped them at all. I think DMD is a great example where we have a gene therapy. It has a microdystrophin because dystrophin's too big to make an open reading frame from. And so you end up with the next best thing that we can invent, and it doesn't in the end work out as well as we would have expected. So the advantage of gene editing is we're fixing the gene that's your gene and my gene that works completely normally, has all the normal regulation, has all the splice forms, all the protein isoforms that are being controlled in ways that we don't even understand right now how the genome really works. We have a relatively superficial understanding of how all these components all fit together and how genes are precisely regulated on a minute-to-minute basis. So fixing the genome back to the normal genome, we predict, and I think a lot of data are consistent with that, it is going to be superior in many settings to a synthetic transgene. So I think that's one element. The other element is that if you fix your endogenous genome back to normal, you don't need to have another gene there. So a gene therapy requires, largely speaking, particularly for AAV, it has to be there in that right concentration permanently in order to work. And if the cell spits it out or silences it, then you will lose efficacy. And certainly we've seen that in the hemophilia gene therapy programs that we've lost efficacy over time. And I think that's also a concern. And of course, then there's the immunological concern about leaving a transgene, a foreign piece of DNA in the body long term. So I think those are some of the risks. They're borne out in some places, not in others. And gene therapies have been very effective in certain areas, and you heard some examples today.
Yeah, thank you very much. Maybe I'll ask Jim, and then maybe Maria, I'll ask you as well, because certainly you folks are developing this whole laboratory of vectors that you're going to be supporting beyond just what you're doing now for Glybera 2, I guess, whatever you want to call the therapy. Jim.
Oh, you're on mute.
2 years of Zoom, and I'm still making that mistake. But someone had to do it, right? I think one of the key issues relates to the suitability of delivery vehicles, and right now viral vectors being the best to get to the most tissues. So I think the area where hopefully there can be some progress is the ability to use non-viral formulations for delivery. Right now, those seem to be confined primarily to a small number of tissues. So for example, systemic lipid nanoparticle delivery really doesn't penetrate skeletal muscle, for example. But you could imagine if you could overcome that barrier, you can now deliver bigger things. You can deliver, endogenous RNA, and you can have overcome the problem of single dosing. There's a lot of potential advantages, but I think that's an area where, and I know NRC is extremely actively involved in the idea of trying to develop new LNPs for targeted delivery. And I think that's going to be an area of real growth. And you think about the editing enzymes, and one thing that maybe, I don't know how much you mentioned that, but like they're too big by and large. I mean, Cas9 barely fits in AAV. Non-viral delivery would really be a huge advantage for many tissues that are now not accessible by non-viral means.
Maybe I just also can comment. Yeah. I think that, yes, I agree with you, Jim. It's the accessibility of tissues that I think is important as well. But I have to say that for us, Glybera 2 had two components. One is that first, there was already, it was opportunistic to a certain extent, in the sense that we knew that there was already a gene therapy that has been developed and that has been successful with no really toxicity observed over the clinical trials. There was obviously efficacy. So the question is, can we make this more affordable? So that was the really incentive of this first gene therapy that we have tried. And as Jim is mentioning, we are also really developing and exploring other ways of delivery, the gene therapy as lipid nanoparticles. And I agree with that. I think it was really opportunistic, but also understanding that it was a Canadian disease, and we had in-house, in Canada, all the participants and all the expertise that brought Glybera to the success.
One thing I want to add is, as important as the mechanism of action, how we repair it, is also getting to the patients earlier in the disease, right? Because if you have microcephaly, you're not reversing microcephaly. If you have axonal death, you're not reversing axonal death. So it also depends on how early we get to these patients. And unfortunately, in most cases, it's before the diagnosis is even done in most countries. And I think that newborn screening has to be really adapted to that, because if we're able to get that earlier on in most of these children or most of these patients, we can make a difference in their lives instead of only slowing down or stopping the disease at most. So I think that's a critical factor, regardless of what mechanism, gene therapy, gene editing, lipid nanoparticle, whatever we're doing, is also extremely important.
Yeah, that's a hugely important point. And Jim made the case before about SMA and newborn screening and getting to those patients, days after, they are diagnosed even before their symptoms. And I think Chris McMaster spoke about that earlier. They're really trying to bring in not only newborn screening, but when do we introduce and what's the relative value of genome sequencing very early on. So I think as we get these methodologies not only more mainstream, but also more cost effective, and actually being able to think about how we treat, I think this will open up some amazing worlds, which maybe I'm really conscious that we're going to run out of time and there's a huge number of questions here, but I'm trying to wrap some of it up together here. Let me this may be for Jim and Terry and Maria more than it is for Jeremy, in the sense that we're talking about, made in Canada solutions. We're talking about, Canadian research. And Jim and Terry, I know you spoke to that deliberately. What's the sustainability model? There's a question here about why these therapies are so expensive, but I'm going to go the other way. Is there a sustainability model for a not-profit approach, for a public-private approach, for a Canadian-grown research approach? Is there a sustainability model here?
Do you want me to go first, or you want to
go first, Terry?
Yeah. So first of all, I want to thank Health Canada for being an extremely amazing partner in my journey. I went through it during COVID, which is the worst possible time ever to go through that, and they were very flexible with working with me and very quick and understanding in my situation. And now that I'm running a biotech, which I can't believe I'm actually running and taking on almost 5 diseases. I think there's an amazing opportunity within Canada. My worry is that, we're only at like 4 or 5 or 6 rare diseases, gene therapies, and they're at $3 million. What happens when we get to 50 or 100? Will our system be able to handle it? And the main problem comes down to manufacturing and the cost of manufacturing, because, it costs roughly $800,000 or $1 million to make the drug. And then on top of that, you have to have, profits and all the other wonderful things that go along with that, and especially if you're manufacturing in pure GMP products. So I think the problem that we're going to have is how do we make a sustainable model in Canada where we're not going to say no because of the cost, right? And I don't think there's an easy answer to that. I think what we have to start doing is getting creative and partnering with companies that are making these therapies early on, so that they will provide us significant discounts, or maybe we partner with them to manufacture these drugs within Canada, and we become early adopters or maybe center of excellence, so that people want to bring them here earlier on, we treat more patients, and then the drug doesn't cost as much because we partnered with them financially to bring them forward. I don't think there's a right answer, unfortunately, because it's just so expensive right now until it goes down.
Thanks, Terry. And I think, as you say, the cost is not only in the discovery of it, and I think everybody's here talked about the many challenges in terms of moving forward with the different kinds of setbacks, but also the actual manufacturing. I think a really good example is the drug for SCID, Strimellus, which was finally given by Orchard to Telethon in Italy, and they're still saying it's $700,000 to make that available, 700,000 euros to make that available to a patient. So it's not for nothing, even when you've gotten all of it, kind of the development costs forgiven already. So it's a challenge. Jim, you alluded to this before, and you also alluded to something that was very scary, and that is that we could make therapies for 1,000 diseases. And I think that's what Terry's saying. What do we do? Is there a sustainability model here?
Yeah, I mean, great. I think Terry touched on the challenges, and I don't think the current model is sustainable, of waiting for industry to come with a $5 million therapy. I just don't see how that we could possibly afford that as a healthcare system for multiple drugs. And so I think then it becomes incumbent on us to figure out, is there a route for a strategy that we do it as a country from beginning to end, and that's going to require coordination between clinicians, scientists, regulators, payers, not something that's traditionally happened per se. And I guess the question I don't know the answer to, and maybe Maria can reflect on this, is even with all that, let's say that there was a subsidized system where we say we're going to make gene therapy for the following 50 diseases. The government's going to guarantee to pay for it if it works at the end of the road. even so, is that just too expensive because of the current cost of manufacturing? Or is there a feasible model where that could fit within the healthcare system? And maybe Maria, you could reflect on the actual costs for your program, because I do wonder, I mean, we hear that it's so expensive, $800,000 Terry's quoting, but is that actually true? And if it is, maybe it's just not ready yet for this sort of mass production. We really need a technological advance in the cost of manufacturing.
Okay, Maria, you're going to give an answer, and I saw Jeremy kind of wave his hand, so I think he's going to give us a commercial gift out of this. Go. Go ahead, Maria.
Well, no, I agree. I mean, the pharma-driven model is not going to work here if it's only like that, because yes, there are so many few patients that you need to treat. This is just impossible. So I think what we were envision is something a little bit more like private, public in relationship, in which we have like a consortium, and hopefully there are some different projects that could be really public goods, in which you have maybe a production cycle just cover the needs for the Canadian population. But in other cases with more commercial incentive, in which you have really more the contribution from the public, so that you can reduce the price to make it affordable for Canadians, and then allow penetration in other global markets where you can, price in a different way, or maybe through some agreements that could be government agreements to reach, the same type of model. I think that at the end of the day, it has to be a public in a private partnership in order to be able to sustain this.
And so before I punt it over to Jeremy, who may almost get the last word out of this, I will come back to kind of, I think that you heard that one of my roles is I am actually very much the chair of the Global Alliance for Rare Diseases, and work a lot in certainly developing countries as well as here. And, we have a real issue, right, in terms of what are we going to do about low middle income countries. I mean, it's one thing for us to say, even in Canada, how do we get access, and can we make it affordable? Once we start to talk about, Malaysia and the Philippines and Africa, et cetera, what are we going to do there? And I think we've been trying to come up with some global access strategies. In fact, we just recently had a good conversation with Vertex in terms of how, with sickle cell disease, as you know, the bulk of the patients are not here. The bulk of the patients are in really depressed countries. And is there any possibility, but knowing that it isn't just shipping, gene therapies down there, there's a whole infrastructure that has to be in place. So what can we do collectively? I mean, but I'll say in Canada, we often talk about gene therapies or these innovative therapies or these, highly, highly effective therapies. To me, it's the answer like climate change. It's the investment. It's the electric car of the future. It's the windmills of the future. We have to invest in these, knowing the technology is very expensive now. But we're going to have to invest in the infrastructure, and we're going to have to invest in getting them to people in order for us to benefit in the long run. So I keep making a plea for the fact that don't think about it as paying for these therapies. Think about it as an investment in terms of what the future of healthcare is going to be. But Jeremy, I'm going to turn it over to you. And even though we hate to give industry the last word, we probably will.
Remember, the people in industry are just the same as the other people who are on this panel. We all derive from the same places. So, I think and I certainly have a great view on the costs of developing these therapies. But I think there's four things I want to say. One is that we shouldn't be thinking about these costs as a cost like a pill, because these are once and done curative therapies that, if they work, they're going to empty out your intensive care units, empty out your hospitals. That's disruptive, and a lot of people are uncomfortable with that because there's an entire infrastructure around hospitals and intensive care units. But you think about the cost of a week in the intensive care unit on a ventilator, that's $1 million. It's certainly more than $1 million in the States. I don't know exactly in Canada, but
I'm going to have to ask you to do this, wrap it up fairly quickly.
Yeah, so that's one area. I think just think about the amortized costs of a curative once and done therapy. And it sounds like a lot, but actually over 10 years, it's very little. The second is, there is a manufacturing cost of goods issue. But I want to point out that the COVID vaccine is an RNA wrapped in a lipid nanoparticle, and it's being delivered to millions of people for very small dollars. So there's a scale issue, and I think if we get the right delivery system, the cost will plummet. And AAVs are more expensive to manufacture, but I think if we can get the delivery systems, we can bring the cost down. The third is the cost of producing the evidence required by regulatory organizations for safety and efficacy. And if we can come up with more expedited systems once we demonstrate safety, I think we could bring costs down considerably. And I think that's a question for us as a community: is how do we get through come up with systems where we don't have to do all the work again every time. And so that takes me to the last part, and that is modularity. And so we as a company, and I think the field in general, is moving towards much more modular platforms where you take our lipid nanoparticle. Our particle's got 6 lipids in it, and it's got three pieces of RNA in it. If we lock 6 of those components and do all the CMC work on those, and it's exactly the same every time you come forward with a new product, you just simply swap out some Watson Crick sequencers in the guide RNAs. They're almost identical products in many, many ways. And so you can think about reducing costs by modularity. You can think about reducing CMC process. You can think about regulatory steps. All of these will diminish and all these will bring costs down. I'll stop there.
No, that was brilliant. Thank you. And I did visit your place in Boston, and I was truly impressed because, as you say, modularity was absolutely built into the design. So we're kind of totally out. Terry, can you do it very quickly?
I'll be very quick. So the first thing I wanted to say is that an average lysosomal storage disease costs the system $170,000 US per patient. So that's a significant cost. You times it by the lifetime of a patient. It's significant. The second thing is, when you talk about manufacturing, if you manufacture a batch of 250 liters, you actually get negative number of vials to be used in patients. And if you start going into 500 and 1,000 liters, then you start getting into usable drug. And I think that's where, as we get more comfortable with gene therapy, we have to start looking at that and saying, can we reuse those vials that may be retained for whatever use and use them after we show that that batch was safe and efficacious, right? Because throwing away 61% on a 500-liter batch is just unacceptable, especially when we're trying to save money and get more drug to patients. So I think those are major things that we need to do and get creative around. How do we release those vials for patients? So
thank you very, very much. I think what was really exciting about this entire set of presentations and panels, it really does show how creative, innovative Canada is in this space and how much we actually will be able to contribute in terms of that global solution, not just for Canadians. And with that, I'm going to turn it back over to Dr. Draper and to Freya to kind of bring us home in terms of next steps.
Thank you very much to everybody. Really great discussion there. So before we leave, and I know people are probably raring to get off, it's been quite a long symposium, but it's been really good. We've had a large number of participants online watching the symposium. So it seems like it's been a fairly good success, hopefully, and we'd like to sort of announce that we're interested in running another version of this event in the future. And we would like to get some of your feedback quickly now on that. And you can see that a poll has appeared. So we just have 3 small questions there, 4 actually, which will get some feedback on the quality of the symposium, but also what we can do for next time. So a next version of this, we're open to suggestions, and the timing of this we've yet to solidify, but we're probably looking at late 2024 or early 2025, maybe around the same time as this one. Whatever the date, we'll make sure that you're notified of this event. We've got your email addresses to the registrations that you made through Zoom for this event. So do leave us feedback. Let us know what we can do. Let us know what topics that we should be focusing on. And with that, I'm going to say thank you to everybody. Real honor to host everybody on behalf of Stem Cell Network and NRC. Thank you very much, and have a great and hopefully enjoyable Valentine's Day. Bye-bye.
Bye.